You go.
Well, good morning, everyone, and I want to give you all a very warm welcome indeed to our Livestock Health and Welfare event this morning. Wherever you are in the country or indeed in the world today, we're delighted you can join with us, and many thanks for participating. I know that we will have farmers, vets, SQPs, students and others interested in animal health research joining with us today, and you're all very welcome. The event today will be recorded, so you'll have opportunity to listen again to the recording, and if you have colleagues and friends who haven't been able to join live with us this morning, then please do let them know that the recording will be available afterwards. So my name is Dr. Philip Robinson, and I'm the Head of Department for the Veterinary Health and Animal Sciences Department here at Harper Adams University, and it is indeed my pleasure and privilege to be the chair for the event today. So this event is a collaboration between the Moredun Research Institute near Edinburgh in Scotland and Harper Adams University here in Shropshire in England. And this is our third annual event, but the first to be held online. So just to give you a little bit of the history of how this event started to take place, the first event was in 2019 when my Harper Adams colleague and good friend Kate Phillips thought that we really needed to do something with the Moredun, and she and Beth Wells from the Moredun were the organisers and driving force behind the very first event held here at Harper Adams in 2019. So we had such an enthusiastic response from everyone that the event was repeated last year, and although we sadly can't meet in person this year to carry on the friendships and networking that's taken place in the past 2 years, the online format that we have today allows us to welcome folks who wouldn't be able to travel to Shropshire, and indeed widens our reach to a much wider audience. But I'm very confident that we will continue to make this a fixture on our calendars every year if possible, because we have enjoyed it so much and get so much out of it. And I hope you get that today as well. Just a little bit of background to the two institutions in case you're not familiar. Harper Adams was founded in the year 1901 as an agricultural college here in Shropshire, and has gone from strength to strength to be granted full university status in 2012, and is indeed now one of the leading agri-food and land-based institutions in the UK, and has indeed been the highest ranked modern university in the UK for the 5th year in a row, according to the Times and Sunday Times Good University Guide 2021. The Moredun celebrated their centenary in 2020. They started in the 1920s as the Animal Diseases Research Association to work in partnership with the farming community to address their needs with respect to livestock health and welfare. The Moredun Research Institute conducts world-class scientific research on the infectious diseases of livestock caused by important viruses, bacteria, and parasites. So working closely with farmers and vets, the Moredun Research Institute strives to improve livestock health and support sustainable agriculture through the development of diagnostic tests and the creation of novel vaccines to combat infectious diseases. Their science also helps to find solutions to other major challenges, such as ensuring safe and sustainable food and water supplies, and the consequences of changing climate conditions, and with the aim of conserving biodiversity. So while both institutions, both Harper Adams University and the Moredun Research Institute, we both greatly value our history and tradition, and we're very proud of that, we are also very much 21st century institutions, leading the way in research and innovation to meet the national and indeed global challenges and opportunities that we have in agriculture today. We have so much in common in our ethos and ambition and I am delighted that we can offer this event to all of you today to give you the opportunity to hear firsthand from fantastic researchers in the field of farm animal health and welfare from both institutions. So we've got a series of presentations today, and we will start with our first in just a few minutes. You should have the programme of events for the day, and we will aim to finish up just before 1 o'clock. And it's important to note that we will have a panel discussion, an opportunity to ask questions of all the panelists at the close of the presentations later on. And you can also ask questions. We will have a short amount of time after each talk just to field a couple of questions if you have a specific question for the speaker just at the time, and you can type those into the Q&A chat, and I will vocalise those questions. So please do sit back and enjoy the presentations, and I hope you find them valuable and interesting. And our very first speaker this morning is Dr. Stuart Burgess from the Moredun Research Institute. And just to introduce Stuart, Stuart leads the Sheep Scab research group at the Moredun, and this group focuses on the development of novel tools for the control of ectoparasites, with a significant focus on sheep scab, which is obviously caused by the sheep scab mite Pteroptes ovis. So recent developments from this group include a commercially available sheep scab Eliza test. It's a blood test which is able to detect infested animals even before the appearance of clinical signs. And it's the first prototype sheep scab vaccine has also been developed by this group, which can significantly reduce mite numbers and the size of the skin lesions. The group are also working on a pen-side version of the sheep scab blood test, which would offer almost real-time disease detection at farm level, allowing rapid treatment decisions and improving farm biosecurity. So we're delighted to have you, Stuart, as our first speaker today, and Stuart's talk is entitled Control of Sheep Scab, What Does the Future Like? And it's over to you, Stuart.
That's you, Stuart.
That's great. Thank you very much, Philip, for that introduction, and thank you for the opportunity to come and talk to you all today virtually. It's a shame we can't be there in person. I would have looked forward very much to the lunch, remembering it fondly from the last two occasions. So I'm going to give you a talk today about sheep scab, and let's just see if I can get that slide to move. There we go. And Philip has very kindly given you the highlights of the talk already, so that's great. You've had a nice introduction to the talk. And I'm going to look at the control of sheep scab and what does the future really look like, you know, where we stand just now in terms of how we go about the control of this pretty terrible disease. So for those of you who don't know, sheep scab is caused by this mite here, Pteroptes ovis. It's an ectoparasitic mite. You can see this picture here, which is by Neil Sargison at the Dick Vet in Edinburgh, and it's a great picture of scab because it covers pretty much all of the classical signs of sheep scab, from wool loss in the foreground here, and you can see the wool on the ground, the sheep rubbing up against fence posts, whatever they can find, trees, barbed wire, anything to alleviate the intense itching caused by these mites. And then here in the background, you'll see this animal who's arching his back and also doing this sort of lip smacking behavior, which is actually really characteristic of sheep scab. So a great picture there that just encompasses all the sort of clinical signs of scab. So it's consistently been one of the top 5 diseases in the UK sheep industry from both financial and welfare perspectives. It's endemic in the UK. I'll come on to costs later, but needless to say that they are significant. About 10 to 15% of sheep farmers seem to experience scab in any one year. Slightly different situation in Scotland, where the disease is notifiable and has been for the last 10 years. And I'll come on to that a little bit later as well. So this is a bit of a close-up of the mite here, and you can see these are the mouth parts. The mite doesn't penetrate into the skin. It's not a blood-feeding mite. It just abrades the surface of the skin, and that causes this serous exudate, which is what the mites then feed on. And the disease is really an inflammatory reaction, an allergic reaction to components of the mite's fecal pellets. So it's largely caused by the mite in order to generate a food source for the mite. These are some mites just on skin here, and you can see that against this kind of like sort of darkened background, it's quite easy to see them, but in reality they are kind of cunningly sheep coloured and therefore very difficult to find if it's a relatively small lesion at the time. The inflammatory response causes that lesion, and that the mites then graze around the edge of that lesion. And what happens is that as that serous exudate that's produced, as that dries out, then the scabs start to form, and that's obviously characteristic of the disease. And they then increase as the mites multiply and move onto naive skin, and therefore the lesions get bigger and bigger. And it obviously is a very debilitating disease, progressive wool loss, and obviously the scab lesion as well. This just two graphs which show the increase in lesion area. So this is lesion area over time. And you can see that there's this significant lag phase at the beginning, and this is where it's subclinical disease. And what happens then is that as the mite population begins to grow, that lesion then starts to expand almost exponentially. And then you can see that that's reflected in the increase in mite numbers. So you have initially quite a low number of mites. They then start to expand at the same time as the lesion expands. And that's because as the mite population expands, they move out onto naive skin, triggering that same inflammatory response. So subclinical disease can last for weeks or even months. And at that point, you've got no visible signs of disease. So once infested, it can take many weeks before those clinical signs appear. But the reality is that during that time, let me just pop this on so you can see some of the mites here just moving about. During that time, that animal is still infectious, and therefore the disease can spread from one animal to another during this subclinical period. And we estimate that between 60 and 90% of a flock can become infested before clinical signs are identified. And then just to show some infested animals here, the mites have been placed between the withers on these animals. And you can see again, this animal doing pretty much whatever it can to try to alleviate the intense itching. And again, you'll see that lip smacking behavior from that animal, and the back arching as well. So let's just move out of that. Yeah, okay, so how is sheep scab spread? Well, the mites can survive in the environment and can remain infected for up to 17 days. But in reality, as soon as they're off the mite, as soon as they've come off of the sheep, their lifespan is starting to decrease because it's not a perfect environment for them. So we say up to 17 days is best to be safe. So they can be spread in the environment from wall plucks, on trees, on fence posts, mites on handling facilities and on transporters, but predominantly it's through direct sheep-to-sheep contact. But the fomite issue, obviously is a big concern for spread as well. There are a number of risks associated with scab. The majority of outbreaks occur between September and March. You can see this just shows the number of positive skin scrapes from APHA over a period of, I think, the last 10 years. And what you can see is that the vast majority of cases occur from November through to March, with a peak in January time, so around now. Elevation, temperature and rainfall are all good predictors of risk. And there's evidence of foci or hotspots, and I'll come on to this in a little bit later, but generally scab doesn't spread that far. So less than 12 kilometre spread within a 5-month outbreak. And that's because of that direct contact or fomite spread. 85% of farms that have reported an outbreak have had at least one each year in the previous 10 years. and 76% of farms reporting no scab outbreak hadn't had an outbreak in the previous 10 years. So once you get scab, it can be very difficult to get rid of it. And that's generally because we tend to make the same mistakes. I'll come on to how we can compensate for those in a little while. There are a number of other sort of perceptions about how the disease is spread. Many farmers obviously associate common grazing with an outbreak of sheep scab. If you take out common grazing, then the majority of farmers either believe it comes from their neighbours or from bought-in animals. However, common grazing is probably the single biggest risk factor for contracting scab in your flock, and it increases your risk by tenfold. So if I come to diagnosis, so this is that picture again, you know, where it's pretty obvious that you've got a problem here. Obviously, you don't know if it's scab. It could be lice, for example. So you would take a skin scraping from that animal. You would look under a microscope and hopefully, well, not hopefully, but if you then found this this mite here, that you can just see this mite here, then you would know that that was sheep scab and you could treat accordingly. But the problem is it's very difficult to accurately diagnose both clinical cases because of the sensitivity of this particular technique. It's not always clear where you should take a skin scraping from. So in these animals here, you could have a pretty good go at it, but in general, it's not always that clear. And then what do you do with animals that don't have any symptoms or don't have any obvious lesions, of course? So also a failure to detect subclinical disease in early infestations. So in another scenario, which is either screening flocks or incoming animals, or where there is no current signs of disease. So this animal is recently infested, it's a susceptible sheep, and it might now take months, weeks before that animal starts to show signs of the infestation. This animal has a current low level of infestation. We could classify it as an immune sheep. It's got the mites, but it's not got any obvious signs of disease, but it still represents a risk. So how do you diagnose the disease in these situations? That's why Moredun came up with the idea of developing a blood test for sheep scab. And this was with funding from both Scottish Government and from DEFRA. And we have developed, I think, now a pretty good blood test. It's very sensitive and it's very specific. It can detect scab within 2 weeks of an infestation. It picks it up before the clinical signs appear. We recommend it's used as a whole flock or management group test, and that involves testing a minimum of 12 animals in a flock or separate group. And we believe that this is quite an important tool moving forwards, and I'll explain some of the uses of this test in a little while. So first of all, how does the test work during a natural outbreak? So on one of the Moredun farms 11 years ago, 12 years ago now, we had an outbreak spread from an infested ewe from another farm. All animals in this study, 159, were examined for clinical and behavioural signs of disease. So here you can see there were 27 animals from the 159 that showed signs of disease. So 132 showed no signs of disease, but in fact 105 of them were positive by the ELISA. So it just highlights the difficulty of diagnosing scab through the presence of clinical signs alone, but also shows that this test can pick up those subclinical infestations. ADAS, 7-8 years ago, performed a cost analysis of the impact that scab can have. This is a hypothetical flock of 500 ewes. Obviously, there was a reduction in rearing percentage by 15%. Then you have the cost of whole flock treatments and also the labour involved in that. So this came to about £6,000 or £12 per ewe, so quite significant costs. If you then separate that out and just look at the cost of treating to prevent scab in the same size of flock, then you're looking at about £900 or £1.80 per ewe. So that's just treating to prevent scab. So from a cost benefit perspective, it looks like it's better to treat to prevent scab than to deal with the costs of an outbreak. However, we know that we can't rely on routine seasonal or annual treatment with, for example, the macrocyclic lactone injectables, because first of all, they're also used for wormer control, and we have issues around resistance. And in reality, good biosecurity and effective quarantine should actually be sufficient. If we then look at the cost of using, for example, the sheep scab ELISA, so if you look at the whole flock testing, we have 12 animals in a management group. We also have the top sentinel testing approach, which is where you use the tups as sentinels, test them before tupping and then post-tupping, and then look at differences in the serological responses. And you can then see if you can pick up scab in that way. I'll come on to that in a little bit as well. So if we look at just the 12 animals in a flock testing, it's about £6 per animal to run the test just now. So your whole flock testing is going to cost about £72. When you put in your vet fees, your call out and so on, it's going to be about £160. Now, compared to the £900 cost of prophylactically treating, so if it's negative, you've saved about £800. And if it's positive, you've added a small additional cost, but at least now you know you're giving the right treatment at the right time. You know it's scab, you can treat accordingly and with confidence. So when might you use that test? Well, for example, instead of routine prophylactic treatments would be a potential option as to when you would use that test. Just on the cost of scab, so I mentioned before about the cost impact, and previously we'd estimated that to be about £8 million per year. But this paper by Emily Nixon, Richard Wall, Hannah Rose Veneer and Leslie Stubbings, which came out towards the end of last year, has now given a more accurate, up-to-date estimate of the costs. And now we're looking between £80 to £200 million per year for the cost of scab. So that's a significant jump in the cost implications of this disease, and therefore more impetus to do something about the disease as we move forwards. So how can we use that ELISA test in control of disease? So we had a look over on the Isle of Mull. They were very keen to do something about sheep scab on the Isle of Mull, and we were equally keen to go and help them. So in partnership with NFUS, we funded a 2-year study using the ELISA to look at the prevalence of sheep scab on the islands. So in year 1, we tested all of the available flocks, 12 animals per group, and what we found was either no scab or very, very low levels of scab. So less than 1% of the samples came back as positive. However, on retest and on clinical examination, where we did get a positive test back, we found no evidence of sheep scab. So there were a small amount of false positives, less than 1% on retest, but we would expect that from this test because it has 97% specificity. Year 2, instead of testing all the flocks, we used this sentinel approach. So we tested the tups pre and post tupping, and this means that we can reduce the number of samples that we need to process. So it brings the costs down. What we saw was a very different pattern of responses to year 1. So more borderline and positive tests, and actually there was an outbreak picked up by the test using the sentinel approach, and we were then able to use the ELISA to follow up any movements from the affected area and limit the spread. Let me just show you the responses for that. So here, this just shows the ELISA responses here for year 1. You can see we had a few sort of borderline, a handful of borderline samples, but actually in year 2, a completely different pattern of response. And as I say, this actually represented an outbreak of scab. However, at this point, there were no further outbreaks detected, but it highlights the need to be vigilant. So just to summarize the situation on Mull, pretty picture of Mull, sorry. We can assume at this stage that Mull is either free of scab. I think that's unlikely, but the scab is probably present at quite low levels. The island status means that the risk of further outbreaks is low, but not completely gone. And in reality, without blanket treatment of all animals and complete biosecurity, then we're not going to achieve complete eradication on the island. So what do we then need to do? Well, we need to ensure that biosecurity and quarantine are as robust as they can be. And how do we do that? So some of the more important things that you can do is to maintain fencing, check for gaps in shared or rubbing areas. Double fencing is ideal, but at least use that to cover those high risk areas. Communicate with the neighbours and coordinate treatments, otherwise you get into this cycle of failed treatments and reinfestation. And select new animals from known sources or health status wherever possible. Treat all incoming stocks or test them, and quarantine for at least 17 or 18 days. Clean any shared handling equipment facilities before and after use if possible. And this little video just shows why biosecurity is so important. So you have here 6 naive animals. None of these have sheep scab. This is Scratchy, who is just coming in now. He has been infested with scab. He's about 4 weeks into an infestation, and you can see quite rapidly that he comes in, starts to contact all the other animals, and actually what happens in the next 2 weeks, all of these animals become infested with scab. So it's really important to maintain biosecurity, because the disease can spread very, very quickly once it's given an opportunity. So because the Eliza has now been running for a few years with BioBest, we now have quite a bit of data available from that. So this data has been analysed by a master's student, Ailey Geddes, who is part of a VMD-funded project that we have running to look at control of sheep scab. So she has taken all of the Eliza data that we had to date, and you can see she's mapped it on this map of the UK here. Red dots are those that have been tested as positive. Blue dots are test negative. And we can start to understand some of the data that we have from this. So this is a summary for the last 2 years of data, just over 500 submissions, mostly as groups of 12 samples. 43% of those submitted have been seropositive. A number of different reasons for submission. 57% were itchy animals, 33% were just for monitoring, 11% were for quarantine, retesting, wool loss or other. And one really important thing that's come out of this, and I just want to reiterate that, is that double fencing significantly decreased the likelihood of a positive sero status. So can't overestimate the importance of double fencing when it's possible, and I appreciate it's not always possible. And just to look at the distribution, so the majority of these are in England, 27% were from Wales, and you know, for a test that's run in Scotland, developed in Scotland, only 5% are in Scotland, and even fewer in Northern Ireland. So is there a reason for that? There are many reasons for that, but one slight complication we do have in Scotland is that, as I mentioned before, the disease is notifiable. So perhaps we need to start to understand if that notifiable status is having an impact on the uptake of the blood test. And if that is the case, then we need to understand how we can look to mitigate that to get better uptake of the test in the future in that area. So what are the main barriers to the control of scab? Well, first of all, the stigma of the disease. You know, it does have a stigma as a disease of poor management. In reality, that's not the case. You know, it is a very easy disease to spread. But that stigma can lead to a lack of cooperation and also to a lack of communication, because people aren't willing to stand up and talk about the disease. So we need to deal with that as a problem. We can't get away from the fact that we don't have many treatment options for the disease. We have the macrocyclic lactone injectables, but they're also relied on for roundworm control, and I'll come on to resistance in that as well. The organophosphate or OP dips, which work fantastically well, but aren't always applicable. And there are non-validated treatments, showers, jetting systems, which have, and I want to reiterate this point, have not been shown to be effective against sheep scab. So this shouldn't be considered as a treatment option. You should be using either an OP through a validated method, which is plunge dipping, or the injectables, whichever is most appropriate for your particular setting. There's obviously the potential for mites to generate resistance to the macrocyclic lactones, and we have published on that already. I'll come on to that in a second. But we also have the potential, if we don't use them correctly, for the mites to develop resistance to the OPs in the future. It is almost inevitable in the future that will happen. And there are no new treatments coming through. Obviously, particularly with the OPs, there are harmful effects on human health and the environment. These are fairly toxic chemicals, so we have to make sure we use and dispose of them in the right way. Obviously, there's a welfare issue around scab as well, and we need to be aware of the impact that can have on the perception of this disease. And there's the cost of continual whole flock treatments, which are often ineffective and are also unnecessary in many cases. Again, do we need to be prophylactically treating for scab in areas that have low risk of sheep scab? So we reported 2 years ago on the first cases of resistance to the MLs. So this was following persistent treatment failures using moxidectin. We looked at 4 farm populations, 2 in West Wales and 2 in Herefordshire. Efficacy of moxidectin was tested using an in vitro assay, and we were able to demonstrate resistance in mites from all 4 farms. So that was back in 2018, and then in 2019 we were not too surprised, but able to show that there wasn't just moxidectin, it was actually to all of the macrocyclic lactones, so we were seeing this resistance. So I mentioned about this VMD funded sheep scab project, and as part of that we've been looking at modelling the impact of transmission and also the impact of treatment strategies. So this is work that's been done by Emily Nixon, who's a postdoc at the University of Bristol. And if we look at the resistance, so even low levels of ML resistance give rise to higher rates of infection. And this I find absolutely terrifying. So a 10% level of treatment failure, so ML treatment failure, will generate a 25% increase in cases of scab nationally. And a 25% level of treatment failure will lead to a 50% increase in cases nationally. This is just modelling data, but it's pretty terrifying when you think of the impact of that. There's some good news when we look at how we can use the ELISA test, then this is a bit more encouraging. So initial results show that this could be a method for national, regional scale control, but only 50% of farmers would need to use the test for it to have a significant impact on the incidence of disease. So some good news to go along with that. And again, we're now carrying out further work to look at the impact that might have on disease. Recently, we were awarded a grant by DEFRA, funded by the Rural Development Programme for England. So this is just starting now and will run until 2023. This is a control of scab based on hotspot hotspot areas within 3 defined regions. So those are the Northwest, the Midlands and the Southwest. It involves the establishment of clusters of farms. We have regional coordinators who are ADAS, the NSA and the Farmers Network. We set up cluster meetings within each of these hotspot areas. We then determine the sheep scab status across each cluster using the ELISA, and then provision of best practice advice, preferably using the farmer's own vets. We'll then perform follow-up visits and further testing if required. And then obviously if we find scab, we will look at coordinating the treatments. But the cost for the treatment is not covered as part of the project. So that will have to be covered by the individual farmers. So one last sort of 2 minutes is control of sheep scab by vaccination. So I mentioned that no new treatments are coming out, but we are hoping to develop a vaccine for scab. So why focus on vaccination as a means of control? Prior infestation, we know of scab, alters subsequent infestation. So if you have an animal, they're infested with scab, they're successfully treated, or the disease is resolved, and they then get reinfested at a later date, the progression of that disease is very different. And that's shown on this graph here. So that shows lesion size with time. The red is a primary infestation with scab. You can see the exponential growth in lesion size. and the blue is a secondary infestation. And what you can see is that there's a significant reduction in the size of the lesions and also the mite numbers. So this is a demonstration of acquired immunity and offers real encouragement for vaccine as a means of control. So we developed a recombinant vaccine, and I'll just show you that there now quickly. So it's a recombinant cocktail vaccine, which uses free proteins from the Myxoroctes ovis. Free injections at the moment. We're hopefully going to cut that down to 2. Those are given 2 weeks apart and with Quillay as the adjuvant. And what we see are highly significant reductions in mite numbers and lesion size. So here you can see lesion size over time. The blue line encouragingly are the control animals, so the non-vaccinated, and the orange line are the vaccinated animals. So that results in a 68% reduction in lesion size by the 6-week time point. So what might be achievable with a vaccine? Well, we know that prior infestation results in a degree of protection, but we're still getting the lesions. We still have mites, but they are fewer and the lesions are smaller. Can we expect to achieve more than this with a recombinant vaccine? To be honest, I don't think we can. We're not going to get sterile immunity. We're still going to see disease, but the lesions are going to be much smaller, and therefore, hopefully, that will impact on the spread of the disease. So what are the effects of that on transmission? And is that justification for vaccination? And in reality, we have to be conscious that vaccine isn't going to be a silver bullet for the control of sheep scab. We have to look at blending that in as part of a control strategy, using the existing treatments, using good biosecurity, using the diagnostic test, and of course, encouraging communication and coordination when it comes to treatment. That's me. So thank you very much for your time. And again, I think as Philip said, we've maybe got time for 1 or 2 questions, hopefully.
Okay, thanks so much indeed, Stuart, for that excellent presentation. And we have one question that's come through on the chat, which I guess ties into your last point. Can some sheep develop resistance to scab, is the question.
Yeah, it's a good question. Resistance, I would say no. They, as I say, they still get infested, but they do. It does seem to be, controlled in that inflammatory response is different. It's not as profound. You don't get as many mites, but they still get the disease. So again, it's really important with a vaccine that we're not going to achieve sterile immunity using that.
Okay, thanks, Stuart. I've got a question for you. How quickly can the test detect scab after infestation?
So it's probably by about 2 weeks. We're detecting IgG, so we're probably looking about 10 days for that to reach, reasonable levels that we could detect using that technology. By about 2 weeks, we would comfortably be able to detect scab. So.
Okay, well, thanks very much, Stuart. I think we'll stop there. Bear in mind, everyone, that we do have a panel discussion and opportunity for further questions of all the speakers after all 4 presentations have been delivered. But thanks again, Stuart. I will now move on to introduce our next speaker. So our next speaker is Dr. Mara Rocchi, and Mara qualified as a vet in 1990 from the Veterinary School of Milan in Italy. And she also obtained her PhD from the same school investigating scrapie. So after 4 years spent at the International Livestock Research Institute of Nairobi, Kenya, working on immune responses to ruminant tropical protozoal diseases, she came to the Moredun Research Institute in 2000 as an immunology and vaccinology postdoctoral scientist. So she's worked at the Moredun ever since on a number of projects, investigating especially the pathogenesis and the immune responses to intracellular pathogens, as well as the immunological consequences of infection during pregnancy. In 2014, Mara began managing the virus surveillance unit at the Moredun, which performs tests, develops assays, and collects intelligence in support of the Animal Health Scottish Surveillance Scheme. And more recently, and in quite a different vein, she's been responsible for establishing a COVID-19 testing team at the Moredun with more than 20 volunteers from amongst the Moredun scientists who are supporting the NHS through daily COVID testing. And Mara's talk for us today is entitled Diagnostic Test Development in Support of Surveillance Schemes. And over to you, Mara. Thank you.
Mara, I need you to share your presentation with me.
Yeah, thank you. I'm just going to do it in a second if I can find, yeah, my tray. And here we are. Is that okay, Blair? Hello?
Yep, got it.
Right.
1 second.
Okay.
There we go. Right.
Can I go? Hello?
Yep, you can go. Sorry.
Okay. Thank you, Blair. So good morning again. Thank you to Philip for the introduction. As Philip said, my name is Mara Rocchi. I manage the Mordun virus surveillance unit at the Moredun Research Institute. I'm talking today about how do we go on developing diagnostic tests in support of animal surveillance scheme. As you can see, there are other names on this first slide. Those are the people that actually do the work. I'm just here talking. Anyway. So on summary, the summary of today's talks, I'll give you a brief introduction to how part of the animal disease surveillance runs in Scotland. I'll introduce you also to the Moredun surveillance unit, and I'll take you through a few examples of development of new diagnostic tests that are being carried out for this reason to support the surveillance scheme. So we normally and traditionally concentrated on diseases of livestock. So we have been working on respiratory disease, abortifacient disease, and about specialized interest in neuropathology. But more recently, we've also moved in different areas such as pig disease, pig-borne disease, and we have been doing some work on monitoring diseases of wildlife. So I will touch not on everything, but on few of those examples. So just to start with animal disease surveillance, what is animal disease surveillance? So there are a large number of definitions of the term surveillance, and the one that I've chosen is the one from the OIE, that is the World Organization for Animal Health. So surveillance is the systematic ongoing collection, collation, and analysis of information, and information is the key word, is data related to animal health, and the timely dissemination of those information to those who need to know. So the action can be taken. Generally, those that need to know are the veterinary authority. They can cascade the information down the line. Surveillance, my purpose is the early detection of disease. But information can be represented by a report of disease in the field from the farmer to the vet, the outcome of a postmortem examination, or the results of laboratory testing. So looking at the origin of this information, we can have active surveillance, where the activity is conducted specifically for the purpose of surveillance. For example, the veterinary authority designs and conducts the activity, and one of those examples is the tuberculosis skin test program. But active surveillance is very good, but it's very expensive. Where there is passive surveillance, where there are surveillance systems when information on disease event are brought to the attention of the veterinary authority without them actively seeking it. So already utilizes what is present in the field, in the lab, in the disease investigation center, and more than participated in passive surveillance systems. So how does passive surveillance run in Scotland? Passive surveillance is a statutory requirement for the Scottish government. Is generally centered around production limiting diseases of livestock and of wildlife, but more recently, some more data has been started to be collated, especially after one of the BVA recommendation on equine, on companion animal, and on food-related illnesses. So the Scottish government funds a collection of information on disease in those animals, on those species, from a variety of sources, which include the SRUC network, Moredun, APHA, and the Food Standard Scotland. And through these, changes in animal disease status are detected quickly and information is disseminated effectively. Basically, the Scottish government provides funding or part of the funding, and we provide and return information to the Scottish government. So how do we do this at Moredun practically? How do we contribute to this? We have a Scottish Government Commission that is run through a tripartite agreement between us, the Scottish Rural College, and the Scottish Government. As a basis of this, we run routine tests such as detection of malignant catarrhal fever or serological monitoring for louping ill, but we also develop new tests when there is a need for those. And we also provide advice to the VIOs, that are the veterinary investigation officers, that are SRUC vets that work in the field. And then again, all these things, all the finding between us, new test development, and the VIOs are again discussed at the tripartite agreement. And the type of tests that we run go from serological tests. We can run ELISA, we can run virus seronaturalization. Part of Moredun is interested in developing point of care tests, like the one that you can see here. And I was hoping that Stu did describe the one that they are setting up for Sheepskin, but unfortunately that has not been the case. We generally run a lot of molecular assays, from conventional PCR to real-time PCR, where you can detect the level of infection as well as if the sample is positive or negative. Sometimes we need to send the test away for sequencing to determine the type, subtype, or genotype of the virus or the pathogen involved. But more and more recently, we're starting to be interested in what we call syndromic assay, where you know that in the specific clinical presentation is not only one pathogen that is responsible or might be responsible for the disease, but there can be many more than one. We want to know who is there, and we also want to know the proportion between those different pathogens. The way in which we carry out this is through two surveillance units. The first one is the surveillance pathology, where we have two histologies, two veterinary pathology, Francesca and Joe, and we have two histology, Claire and Lauren. The pathology unit has traditionally been concentrating on neuropathology and fetal and maternal pathology, although more recently we have started to expand the type of work that is done in the unit. They also carry out test development. They support the Scottish surveillance and the Moredun researches, and they work in close collaboration with the SRUC pathologies. There is a knowledge exchange component in what they do. There is a 36 to 48 turnaround time for sample processing and reporting, and they normally process approximately 1,000 samples per year, 900 of livestock and approximately 100 of wildlife. The second unit of Moredun is the virus surveillance unit that I manage. Maddie is our lab manager. We have Ellie and Dylan that are our research technician. Holly is our PhD student, and Holly is also a Harper Adams graduate. She graduated with a BSc in bioveterinary science in 2012, so we're very happy to have her with us. And Fiona is our admin assistant. In the virus surveillance unit, we have containment 2 laboratories, and this is just a quick shot of what you can see of the containment rooms in the labs. And we do carry out continuous development, the refinement or diagnostic technique for endemic disease of livestock and wildlife. We run routine and on-demand specialized serological, molecular, and virological tests. We carry out knowledge exchange, and we have to adhere to strict quality assurance schemes. But we also provide advice and support to both Moredun researchers and the VIOs. And not directly linked to the virus surveillance unit, but because we have within our unit and within other units of Moredun a lot of expertise, we have containment level 3 laboratories and animal unit. And this is just a quick picture of the containment level 3 lab, but I'm sure that we're currently using for the COVID testing, but I'm sure that Lee will expand much more on this. So how do we go on developing tests for surveillance activity? There is a logical process, and I'll show you some examples. So we start with an observation. It could be in the field, could be in the farm, could be a re-emerging disease, it could be a novel disease. So we then discuss between us, SRUC, and the Scottish Government if there is a need for a new test development. And if we reach an agreement, I do start with the literature analysis. I want to gather as much information as I can on the disease, and also if there are already tests that have been published, because something has been published and working, there is no need for us to restart from the beginning. They will save us time and money. So one of the two ways I will try to get as much information as I can. We choose then the assay. Do we want a serological assay? Do we want a molecular assay? Do we want both? And once this has been decided, we start with the initial laboratory development. It is a laboratory validation. It's not only proving that the test is working, it also involving gathering positive and negative control samples, in identifying the specificity and sensitivity of the test, and making sure that actually the whole package, the whole test package is ready to go. Once we have successfully completed the laboratory validation, we move to probably what is the more important step, that is the field validation, and this is done in collaboration with the SRUC veterinary officers. That means we check the test with sample coming from the field in real time, because we need to make sure that the test is not only working, but is also robust enough to cope with all the possible combination of disaster that you probably know can happen when samples are sent, collected at postmortem and sent from the field. Finally, if the test passed the field validation, we run the test as a routine test or on demand. And if the volume of the test is too high for our unit, that is quite small, or there is a commercial reason for it, we can transfer the test for SRUC that can run it much more extensively. So I will use this arrow throughout the example that I'm going to show you to indicate where we are with a few tests that I'm going to describe. The first one I want to talk about is the new test that we have developed for bovine respiratory disease. As you probably know, bovine respiratory disease is a multifactorial condition. We have predisposing factors such as nutrition, stress, transport, or a lower immunity that allow viral insult to take place, and generally after the initial viral insult there is a secondary bacterial infection. So as you can see already from those three points at the bottom, you have more than one possible pathogen involved. In fact, the pathogens that are normally recognized at the basis of bovine respiratory disease, they're not all of them, those are just the most common, are bovine herpes virus 1 or IBR, bovine parainfluenza 3, bovine respiratory syncytial virus, and bovine coronavirus, that are followed by Mannheimia hemolytica, Pasteurella multocida, Staphylococcus somni, and Mycoplasma bovis. Mycoplasma bovis is an exception because at times it does not need the viral insult to start the disease. So as you can see here, you need, what is the syndromic assay? You need to test for all the pathogen together. Your assay has to be quick and has to be not too expensive, otherwise the farmers will not be able to afford it and cannot be rolled out in the field. So we started to look at how to carry out this assay. We took advantage of having that liquid handling system that you can see on the left-hand side of the screen that was already present at Moredun through a collaboration with a different group. We spoke to the supplier, we identified the target, and the supplier did the initial bioinformatic design and the design of the assay. That we call it V1, version 1. So using this liquid handling system, what we perform is, for the one that on you that know a little bit of molecular testing, a nice two-step real-time PCR with CyberGreen. What that means is that it's a specific test because you do a molecular amplification twice, and the second one up to nest inside the first one, so only very specific reaction can take place. Is a two-step test. That means you increase the sensitivity. It's a real-time polymerase chain reaction. That means they give you a quantitative or a semi-quantitative indication of the amount of pathogen that is present in the sample. And using the CyberGreen technology means we can keep the cost down. The assay is designed to go up to 16 possible targets. We have selected 11, of which 3 are controls. So the first thing that we did, we validated on reference strain and on clinical samples in the lab. And we quickly went from version 1 from version 2, where we did a bit of redesigning of the assay. And now, using version 2, we have obtained the level of sensitivity and specificity that we require. The assay has been tested in parallel with the previous respiratory assay, and we have shown that it's more sensitive than the previous test. Has a similar price to other molecular tests and is currently under field validation. And this assay will quickly be transferred to SRUC. The transfer was supposed to occur last year, but the pandemic has been delaying this type of transfer. And this is just to show you what happens. The assay is also automated, so you get a screenshot at the end of the test. You can see here that there are, well, you probably can't, different pathogens. They're listed here. It will ask you to check if the pathogen is present, or will tell you if it's negative. Each one of those cards correspond to a different target, and those are the positive tests, and the ones that are negative will stay on this bottom line. And also we check for specificity with an additional step that is represented down here. And so for this assay, we went in between 2 or 3 years, we managed to go from the initial concept of the assay to what is now field validation that is almost concluded and is, we're writing up the paper, and to routine testing of respiratory sample in our lab. Another assay I wanted to show you is development of a test for tick-borne fever. Tick-borne fever is condition caused by a bacterium called Anaplasma phagocytophilus, and this bacterium causes immunosuppression and abortion in sheep, but it can also affect a large number of other species, including humans. The bacterium invades the white blood cells, and you can see here those are granulocyte neutrophils. These are the nuclei of the cell, and this little colony here, this is the bacterium. This immunosuppression, because the bacteria target the cells that are responsible for the immune response, can quickly lead to severe manifestation of disease. The most severe is louping ill, but we can also have severe pneumonia, articular problem, abortion, etc. The diagnosis of tick-borne fever is important because this can influence the prophylactic and therapeutic approaches such as tick treatment, tick prevention, or the use of antimicrobials. So there was a diagnostic test in the UK, but that was lost in 2011. That was a serological test. So in 2014, when I started to work in the virus surveillance unit, we discussed trying to re-establish the previous serological test. But the previous serological test, when we started to look into it, at disadvantages, it needed the bacteria grown in tick cells. And here with the arrow, you can see the colony of the bacteria that is growing in the tick cell. This is the tick cell, this is the nucleus of the cell. It's a very cumbersome, it's a very long and not particularly successful process. The test had a difficult interpretation, and so while we haven't discounted the idea of developing a serological test, we decided to go quickly and develop a molecular test. And we were lucky because we found published methods that have been very effective and we could use and establish very quickly. So since we started testing with a molecular test, we have detected a much larger number of outbreaks, not positive animals, every year. That was in blue here on the left. You can see the outbreaks that were detected or confirmed with the previous test, where on the right-hand side in red, you can see the number of outbreaks that we have managed to confirm in the field since we started with the test. The test was then developed and finalized at the end of 2014. We have tested more than 1,500 animals since we have reached positive diagnosis in 40% of the cases. We have managed to validate it from many sample types that allowed testing live and post-mortem animals. We have a 1-week turnaround time, and the test has been routine available to SRUC since 2016 and been adopted by a PHA in 2017. So for this test now we went quickly, well, in 3 years, 2 to 3 years, we went from the concept to field validation, and now we are on routine testing. The next one, I want to move to the wildlife. Part of the work that we do with wildlife testing involves testing grouse sera for louping ill antibody. Louping ill is a viral disease that is very detrimental to grouse chick. Is up to it can cause up to 90% mortality in very bad years. So the collection of this sera that are used to monitor the grouse and the moors is done in a what I would call cumbersome, unsafe way. It requires expertise and requires dedicated personnel. As you can see here, the grouse are taken after a shoot enough is inserted in the chest, and then a glass capillary tube is inserted in the hole to collect the sample. And you can see you have a glass capillary tube that's very thin, and you're working with a potentially zoonotic disease. So when I was looking at this, I also came across a paper where they were using a different collection system that is based on this, that is a filter paper collection system. The method has been tested and validated in other species for serological assays. So we decided to go and try to see if we could use this collection method for the collection of the sample and the testing in our lab. So this is me actually, that insert the filter paper inside the chest of the grouse, and that's what the collection works. So when we ran the test in the lab, and I ran it by myself, the test, the sample collected in this way performed well. Although once we started to send the filter outside to other people to do the collection and more critically the drying of the filter paper, we find out that the test did not work particularly well. So once we thought that we were on the file validation stage, we actually have to rethink again. And probably it's an interference of the collection method with the test for the detection of the antibody that we run in the lab. We are developing an Elisa, and Elisa is supposed to work very well. So sometimes we get to the point that we are at the field stage, and then we have to rethink again. And the last example I want to give you, if I got a few more minutes, is some work that we have done for, again, for wildlife, but also for for pets and breeders on Rhdb2 Diagnostics. So what happens is that we were called at the end of 2014 to assist with the diagnostic for a large breeding colony of rabbit. They had 1,500 animals per year, and they started to have sudden deaths in various rooms. Over 11 days, they lost 30 animals, over 200. Was in a hyperacute form with few clinical signs. The animals were in good body condition with neurological symptomatologies appearing. And the vet that assisted the colony had the suspicion of Rhdb. That was a virus, a rabbit hemorrhagic disease virus, that was known to be present in the UK. So I spent an afternoon on the phone trying to find a lab that did a serological test to confirm disease, with no luck. So at that point, the people running the colony were becoming a bit desperate. They needed to know what was going on. So myself and a colleague, we picked up the OE manual, we looked at the protocol. We found 2 set of primers that we could order for the testing, and we sent off the order that came back 2 days later. 2 days later, we were able to run the molecular test. This is a classical PCR. And as you can see here, this set of primers called RHDG did work very well. It gave a nice positive band of the molecular weight that we expected, but the second set of primers we had to discontinue because the condition that we were using at the time did not work particularly well. Although our problem is that we have no positive control samples, so we had to purify it and send away this nucleic acid to have a sequence, to confirm actually the etiology of the disease. The consequent sequence came back and it said, yes, it's Rhdb, but we also realized when the sequence came back that we found the first detection of a variant of Rhdb that is called Rhdb2 in Scotland. And more or less at the same time, a bit earlier than us, but not very much, AHVLA at the time, but now APHA, had published that there was a Rhdb variant 2 starting to appear in England. And so we also put information out there. This is part of our knowledge exchange duty to alert the vets, but alert breeders and pets owner of the presence of this new variant of virus. And the problem here is that the vaccine that was available for the classical variant would not protect from this virus. So we were here. We did run the test as a routine test for 2 years until the commercial laboratory caught up and developed their own assay. So we still keep running the test for wildlife cases, but we now transfer requests of breeders and owners to the private laboratory because this is not really our remit. And finally, we have a number of tests in development. I hope I'll be able to come back another time to tell you how are we progressing with those. We're looking at developing we're validating this one just now, a cross serum neutralization test to distinguish response between border disease virus and bovine diarrhea viral virus. we are looking at using nanopore technology, and this is next generation sequencing where you can detect more or less everything that is in your sample, not only what you think is there, but what you can and you could detect with normal tests, but also pathogens for which you don't have specific tests. And we are starting to establish a syndromic multiplex abortion PCR. This work again was stopped last year due to the pandemic, and my attention moving to the coordination of the COVID testing. So I just need to acknowledge a lot of people have been involved in this work, and I'm sure that I forgot many of them, and I'm happy to take any question if there is any time.
Well, thank you very much indeed, Mara. Thank you, Mara. So that's a very interesting insight into laboratory science and development of diagnostic tests, and stressing the importance of having tests available to cope with the disease demands that are out there, both in livestock and in wildlife. Questions, if you're not familiar with how to see the questions and answers, there's if you hover your mouse over the screen, there's a little two speech bubbles with a question mark on them. If you click on that, then you can see the live Q&A coming up on the right hand side of the screen. So please do feel free to ask your questions there. Mara, can I ask you, how long does it typically take to move from evaluation of the need for a test to the final validation of a new test?
That depends in part of the type of test. Molecular tests are much easier to develop and establish the classical serological test or tests that require, for example, growing the virus. And also depend on the availability of samples. That is the important things that I would like to stress here. Is not ourselves in the lab doing the work. We need to work in collaboration, close collaboration with the vets from the veterinary center, the farmers, but also the private veterinarian, because more samples we have with specific clinical history and indication of exactly what was a suspicion of what is wrong, and more quickly we can move through the validation of tests. So it can take a year. If you are very lucky, it can take 4 to 5 years. And the COVID is a very good example of COVID-19, because people that look at establishing the test were able to establish the test because they had the resources and the need to do it in less than 2 months, and that was brilliant.
And you mentioned at one point about spending time on the telephone to find out if there was a test for, was it RHDV?
Yeah.
How much importance would you place on those personal networks and knowledge and finding tests and developing new tests, the network of scientists, and how important is that?
It is. The network are really fundamental, because that's probably one of the early way we can start to understand that there is a problem on the field. And again, it's not only scientists, it's vets, it's farmers, it's whoever can bring to us or to the veterinary authorities observation that sometimes might seem a bit strange, but at times are really important. And I think an example of this one was Schmallenberg, when it first started, and the dairy farmers started to realize that they were having fetuses that were not what they were expecting, that there was a much higher incidence of the disease. So yeah, but we do keep collaborating with a lot of different scientists and a lot of different practitioners, and people phone us if they want to. We're part of the Moredun Foundation, has a large number of members, is over 10,000, and we are happy to answer questions within reason. And there is one of our receptionists, they're very good at directing the question to the correct person. So please feel free to contact us if there is a problem or if you want to discuss something.
Okay, thanks so much, Mara, and thanks for your excellent presentation. There will be an opportunity to ask further questions of Mara and Stuart in the Q&A session at the end of this morning. So it's time for our 10 minutes refreshment break now, so please do have a break and come back and join us at 11:20 when our next speaker will be Professor Lee Ennis. So 11:20 and we'll start sharp again. We're on time, and 11:20. See you soon. So it's now 11:20, so we will make a start again. So welcome back, everyone. And our next speaker is Professor Lee Ennis. And Lee is the Principal Scientist and Director of Communication at the Moredun Research Institute. And she has an honours degree in immunology from the University of Glasgow and a PhD in tropical animal health from the University of Edinburgh. She's really interested in the relationship between the many pathogens we encounter and our immune responses, and how this can be used to develop better methods of disease prevention and resilience. She spent her early career working in Morocco and Kenya, and then took up a position at the Moredun to research vaccines to prevent and control livestock diseases. And she's very interested in adopting interdisciplinary approaches involving collaboration across different sectors, and also in science communication and education. And Lee was awarded an MBE for services to scientific research and science communication, and she is an elected fellow of the Royal Society of Edinburgh. And Lee's talk today is adopting a One Health approach to tackle zoonotic diseases. Over to you, Lee.
Well, thank you very much, Philip, for the introduction, and it's a real pleasure to be here today to speak to you. Like the others, I'm very sorry we're not able to do this in person, but it's great to be here virtually. So what I was going to do is discuss with you over the next 20 minutes is looking at a One Health approach to tackle a range of different zoonotic diseases. And I'll pick up some examples of some of the work we've been involved in at Moredun. But just to give you a sort of concept of One Health, it really is a sort of collaboration and coordination between various different sectors, looking at human health and public health, livestock, wildlife, companion animal health, and also the environment. So essentially it's looking, as it says, on looking at all of those aspects and bringing them together in a coordinated way to look at achieving the best health outcomes for people and our planet and animals. So very pertinent at the moment, as we're in the middle of the COVID pandemic, which has hit everybody across the world. And I'll touch on that a little bit as we go through the talk. So just to give you an introduction, a bit of background, what we're talking about, we're looking at zoonotic diseases, and these are the ones that are transmitted from animals to people. They can involve viruses, bacteria, parasites, and they're spread through very different routes. It's a major global health problem, and this is really due to, we've got many people on the planet, as you probably know, and we've also got very close association with animals, either in agriculture or companion animals. So if you look at some of the stats, and this is coming out of OIE and WHO, about 60% of the existing human infectious diseases are zoonotic, and 75% of those of the emerging diseases have got an animal origin, which just gives you the major context of zoonosis and adopting a One Health approach. 20% of loss in production globally from food animals is also due to disease. So this is giving you a really large scale context of why it's so important to understand infectious disease and look at various different ways of tackling it in a holistic way. So what I'm going to do in the rest of the talk is pick up some examples of the work we're doing at Moredun in zoonoses, and give you some different approaches that involve a sort of One Health take on this. So as Mara has talked about earlier, at Moredun we've got specialised facilities and highly trained staff to work on high hazard group pathogens, and these are from containment level 3 and also in containment level 2 and below. So we can work on many different high hazard group pathogens. So the ones we're focusing on a bit at Moredun are the ones shown there, which are E. coli, Campylobacter, Toxoplasma and Cryptosporidium are some of the main ones we're looking at. Now, I know that Hannah will be following me and she's going to focus a bit on Cryptosporidium, So I'm going to pick up examples of looking at E. coli, Toxoplasma, and I'm going to finish a little bit to tell you about some of the work we've been doing on COVID. So start off with E. coli. This is one you're probably very familiar with, and it's an interesting one because the E. coli bacteria are generally harmless residents in the gut of cattle, but you've probably seen a lot of information in the press and a lot of cases of some very virulent strains of E. coli 0157, which causes severe diarrhea, kidney damage, and this is largely due to the Shiga toxin, which is in the strain of E. coli. And those of us that live in Scotland, there is a very high prevalence, unfortunately, of this strain of E. coli in Scottish cattle. And interestingly, surveys done a few years ago, if you looked across the UK, about 90% of the cattle in Scotland were shedding this bacteria, compared with other parts of the UK. So Scotland got quite a bad reputation for E. coli. Now, if you look at this from a kind of One Health perspective, the issue is probably not for the cattle themselves, but the issue is a public health problem. If the bacteria is transmitted to people, it can cause very severe disease, as you've probably heard of. Some foodborne outbreaks have been very prevalent. So the challenge we had at Moredun was to try and develop what we call maybe a food safety vaccine. So this was a way to really protect public health by vaccinating food animals to protect against the zoonotic infection. And this is work done over a number of years by Tom McNeely and his group, and also in collaboration with the Roslin Institute. And to summarise that quite quickly for you, what Tom and his group did was they looked at different components of the bacteria that allowed the bacteria to attach to the gut of cattle. And in doing that, they developed a vaccine which had two components. It was the flagella component, and I can see a picture, if I can show you there, of the bacteria. This is the flagella. This is how they attach to the gut. And the other component that the group looked at was a type 3 secretion protein. So both of those together allowed the bacteria to attach to the gut of cattle. And then what the team did at Moredun was, because we have the high hazard group facilities that we can work with these bacteria, is Tom set up a challenge trial at Moredun, and this is the advantages we have there of being able to work with some of these pathogens, and looked at efficacy of the vaccine. And what they found was that the vaccine had very high efficacy in reducing both shedding of E. coli and also transmission between cattle. So quite excitingly, this vaccine is now undergoing commercial trials in the USA with feedlot cattle. And it's a really good example, I think, of One Health approach. We're looking across the sectors on both the veterinary side and the public health side to develop a vaccine to help protect people. So I'm going to move on now to talk about a parasite that I've been involved in researching. So I'm going to spend a little bit more time on this one. But it's a very interesting one. It's a parasite called Toxoplasma gondii. And as many of you know, the cat is very important in the life cycle of the parasite. I've got a diagram here just to talk you through this or remind you a bit of the life cycle. So cats get infected by eating rodents or birds that are infected with toxo. And the cat, interestingly, is the only animal where you can get the sexual life cycle of the parasite. And they shed these oocyst eggs, which are very resistant in the environment. And that stage can cause infection in both livestock and with people. And the consequence of infection, I'll start with the livestock, is if sheep get infected with the parasite from the first time when they're pregnant, it can cause abortion in the lambs. So it's a very important cause of infectious abortion in the livestock industry. Similarly with pregnant women, if they get infected for the first time with the parasite when they're pregnant, it can cause problems for the developing fetus. So it's a very serious disease in people as well. And the other route of transmission I wanted to highlight to you, because of the One Health side of things, is that many food animals can get infected with this parasite. And what happens is if we eat meat from infected food animals, that's another way we can pick up the infection. So over here on this side, I just want to show you pictures of the parasites. So this is the oocyst stage, not as big as this. You can't see it in the environment. They're very, very tiny. You can get about 100,000 of them in the head of a pin, so you can't see them. This is the parasite developing in the cells. You can see these small sort of green banana shapes. That's the toxoplasma developing inside our cells. And this is where it encysts. So this is what you'd see in tissues that are infected with the parasite. Again, not to scale, these are tiny, but you can see the parasite absolutely packed in to the tissue cyst there. So if I look at just talk you through the impact of this disease, it's a major cause of infectious abortion in sheep worldwide, so not just in the UK, across the world. It's the second most important cause of infectious abortion in the UK. And that point about the parasite cysts do remain in the tissues of sheep. And this is not just sheep, it can be pigs and cattle as well. So chickens, all food animals can be infected with toxoplasma. So it's a problem if you enjoy rare or undercooked meat. This can be a problem for transmission. So there has been some work recently which has highlighted the importance of toxoplasma as a foodborne pathogen, which brings in that One Health side. We've known for a long time it's a problem with abortion and infertility in sheep and goats, but interestingly it has now come to the fore as one of the most important foodborne pathogens as well. So if I just talk about the disease in sheep first of all, this is mainly what we've been involved in at Moredun over the years. And we've done some work looking at trying to understand that host pathogen interaction with the idea of developing a vaccine to protect sheep against abortion. And if you can see the picture up here, this is very typical of what you'd see with a ewe that gets infected in mid-pregnancy. You get a stillborn lamb here, and I don't know if you can see that, it's a little mummified fetus just beside the placenta there. This is very, very typical of toxoplasma. And the major problem is if the sheep gets infected for the first time during pregnancy. And down here, if I can show you the placenta, again, this is very typical of toxoplasma. The placenta in ruminants, you get this cotyledon structure, and this is where the mother and the fetus are sort of joined during pregnancy. And if you can see there's lots of little white spots on that. And that is essentially the mother trying to get rid of the infection in the placenta, and she's actually causing a bit of pathology here. So in her trying to fight the infection, she's compromising the placenta, and that often leads to the abortion. So we've looked closely at Moredun as to what the outcomes are if sheep are infected early, middle and late, and you tend to get more severe disease early and mid-gestation with this parasite. And interestingly, this is very similar to what you see with pregnant women. So the disease in sheep in terms of congenital infection is very similar with people as well. Now the good news is following infection, sheep do develop very good immunity, which will protect against disease going forward. And this is what we exploited to try and help develop a vaccine for toxo. So this was a live strain of toxoplasma that was originally isolated in New Zealand, big sheep industry in New Zealand. And this was, interestingly, it was an incomplete strain, which means it only is the fast multiplying stage of the parasite. So this is the tachyzoite stage here. So the parasite will not differentiate into a cyst. So it gave an opportunity of a live vaccine. This was originally developed in New Zealand, and we did the work at Moredun to try and do the efficacy testing so that it could be launched and used in Europe. So the experiment we've got here is a very crude one, but just shows that when the ewes were vaccinated prior to tupping and then challenged with a strain that we knew would cause abortion, if you look at this one here without the vaccine and challenged, we got 10% viable lambs. With the vaccine, we got 80% viable lambs. So the vaccine was highly effective in these challenge models. And again, we were able to do these experiments at Moredun because of the facilities we have to handle those high-level pathogens. The immunity duration was 18 months, which was very good. So it meant we could keep the duration of immunity was very good in terms of a one shot of the vaccine. We did some work early on, just look at the immune responses that were generated by the vaccine. And because it's a live vaccine, it's probably not a surprise, it was mainly CD8 and interferon gamma, which were important in protective immunity. So this is the only commercial vaccine available worldwide for toxo, and it's supplied by MSD, but it is a live vaccine. There are some safety distribution problems with it because it has to get into the animal very quickly, so it's got a very short shelf life. It's highly effective though, and it's mainly because it's live. So the challenge we've got here is to try and get something that has maybe got a longer shelf life to make the distribution a bit better. But the challenge we've got is to stimulate the right immune response, because it does require that cellular immune response. So if we look at part of the sort of One Health area is to really look at that sort of interface between the veterinary side and the human side. And this is usually the point of the talk where I can't see you all in the audience, but I suspect a lot of people who might be tuning into this might well be infected with Toxoplasma. We did some work in Scotland, and just to give you some of the prevalence data there, we looked at blood donors in Scotland. So these are people aged between 20 and 60, and we looked at the seroprevalence and found that about 12% of people had antibodies to Toxoplasma, suggesting they had been infected with the parasite. And this is not far off what we think worldwide. There are about 1 in 5 people are infected with this parasite, so it's very prevalent. We also looked at, we got some samples from a sudden death brain bank in Edinburgh, and we looked directly in brain tissue and found that the prevalence was 18%. And again, it just shows that the high prevalence across Scotland. We've been working with colleagues in Brazil, because in Brazil they've got, in South America generally, they've got some very virulent strains of the parasite that seem to be a lot more pathogenic than the strains we're seeing in Europe. And I think it's very important and pertinent that we do understand that some of these strains can change, they can become more virulent, and therefore some of the work in surveillance becomes very important to understand what is happening across the world with this parasite. And again, looking from a One Health perspective, it's interesting to know is what is the major route of transmission to people? And looking at risk factors, it seemed that the most important transmission route to people was this consumption of undercooked meat from food animals. So if we look at this as a foodborne pathogen, and this is an interesting sort of analysis of the impact of some foodborne pathogens, because you've probably heard of the incidence of Campylobacter is extremely high, same with norovirus. And if you look at that graph there at this side here, where we're looking at the impact, there is a way of measuring impact called disability adjusted life years. And this is just looking at the impact to people if they become infected with Toxoplasma. And if I refer you back to the main issues really are pregnant women that get infected for the first time, and it can cause real congenital problems in their children that are born. They can be blind and deaf and have brain damage. So there's a significant impact of this parasite in people. So while the incidence may not be very high, the impact is very big. And that's why it emerged in the last few years as being a major problem by both the WHO and the European Food Safety Authority. And we did a paper with them, we worked with them a couple of years ago, and they produced a scientific opinion looking at the public health risks associated with foodborne parasites and highlighted the importance of Toxoplasma. So what can we do about this? So if we were looking at a One Health approach to vaccines, and this is what we're very interested in at Moredun, if you look at what your key targets are and how you might go about it, we produced a paper last year discussing approaches you could take to vaccination for toxo. And the vaccine I talked to you about before, the S48 vaccine, will prevent congenital disease due to Toxoplasma in sheep. There is currently no vaccine for people. And it's something that I think people are very interested in looking at now due to this analysis that, and especially in South American countries where you've got these really virulent strains, a vaccine for people would be extremely important, and it would be targeting congenital disease in women. The other route we have done some work on is to vaccinate food animals. So we talked before about the risk from food animals. There is slightly different risk depending on the animal. The most problematic one seems to be outdoor reared pigs. They do seem to have more cysts than, for example, cattle. Having said that, we are more likely to eat rare steak, and therefore, if you look at the risk of it, actually beef has come up very, very high. So we've done some work looking at using the same live vaccine that we've used for protecting against congenital infection to look at to see if we could, like a food safety vaccine, could we protect pigs and sheep using the same vaccine to reduce cysts? And that vaccine did work. So the live vaccine will also protect against tissue cysts in food animals. The big challenge we've got, and we're not doing any work on this at the moment, but other people are across the world, is to look at, could you vaccinate cats to reduce oocyst shedding, because that would make a huge difference to the transmission of the parasite. So just looking at summarise the One Health approach here for Toxoplasma. I said at the beginning that the concept of One Health is really to bring in lots of different disciplines, lots of different opinions from people, and lots of different science from different sectors to really tackle the disease. So with Toxo, one option which we've been looking at a lot at Moredun is the vaccination option, and we've discussed maybe the different targets looking at the transmission of the parasite. The other thing people have looked at is biosecurity around livestock farms to try and keep out the parasite. That's slightly more tricky because there's, I haven't shown you the data today, but there's a huge amount of environmental contamination with Toxoplasma oocysts. So cats are really a problem here, and these oocysts last for a long time in the environment, so it's quite difficult to clear the parasite from the environment. Lots of work going on in diagnostics and detection, and this is looking at contaminated food and water, looking at parasite strain variation and virulence, and we've got a new grant at the Moredun, which Claire Hamilton is leading, looking at virulence of the parasite. So the other thing is, I haven't talked about, is things about consumer practices. So what can we do? If you freeze meat, you will kill the cysts. If you cook meat, you will also kill the cysts. So there's various things one can do to help tackle the parasite from various different angles using a One Health approach. So I'm going to finish up in the last few minutes just to discuss with you something that Moredun has been involved in recently, and something that Mara touched on in her presentation. But if we go back to probably this time last year, I think the first COVID cases were detected in the UK, and come March, as we all know, we've all been in it, there was that first lockdown. And during that time, we got together, a few of us got together at Moredun, and we thought, well, look, we've got the facilities, we've got the staff to do, as Mara was describing, the high throughput diagnostic together with SRUC. Could we help at all with this pandemic? So what we did was we got in touch with colleagues at the NHS and offered our help in terms of diverting some of the veterinary diagnostic to help with the NHS PCR diagnostic, which has been done at that time. I've got a graph there at the bottom, which you can see. I've just lifted this off the BBC website, but it basically covers the detection of cases in Scotland. It's probably reasonably similar, a bit more down in the UK, but across the world you've got that pattern. Thankfully it's going down a bit at the moment. I hope that trend will continue. So what we did to set this up was we contacted the NHS, we described what facilities we had, and then since then we were working together with SRUC and the NHS to set up an end-to-end testing facility at Moredun. And this is for the RT-PCR test to detect SARS-CoV-2. And this was a huge project involving a lot of people, but it involved establishing SOPs, protocols, validating the test, proper quality control. We had to repurpose some of our laboratories at Moredun. We had a fantastic response from the staff, and Mara and her team have trained well over 20-25 staff now, and some of them are volunteers from the Institute, which was absolutely fantastic. Then we had to set up the IT infrastructure with the NHS, because this is an end-to-end service, so it's basically like the NHS at Moredun. So the testing node went live in June 2020, last year, to support the NHS. And we were very proud that the Moredun SRUC node was the first veterinary testing node to be set up to support the NHS, which is, I think, is a fantastic example of One Health in action. Now, to date, we've tested over 21,000 samples from hospitals, care homes across Scotland, and we've also taken part in helping the NHS with some research for some new testing developments. So looking at pooling of samples, this is something that SRUC do regularly with the BVD testing. Also looking at the effect of collecting samples in different media. And the other thing we were involved in recently is, you probably know that one of the issues we've got with the COVID at the moment is the virus does mutate, and there have been some new variants that are coming up. And the test we were actually using, the PCR test we were using, was actually able to help in the diagnosis of the new variant, because it targets 3 genes. And one of the genes, the S gene, was dropped out with the new variant. So using the test we had, we were able to help identify cases across Scotland of the new variant. So just to say, well, I'm finishing up now, but really just to say, this was a really good example, I think, of people working together across the sectors. Sheila Vos, our Chief Veterinary Officer, was delighted with the collaboration, and Ingo Johansson was our main contact at NHS, and I have to say he and his team are doing a fantastic job dealing with a very difficult and continually changing situation, and it's been great to be able to help with that. I'll finish up there just to sort of emphasise that One Health is a concept that we use a lot these days, but it's a really important concept in dealing with these zoonotic diseases, and essentially involves really good collaboration between sectors and communication to get good outcomes. I've got masses of people to thank here. I'm not going to name them all, but really just to say thank you very much to some of the colleagues at Moredun whose work I've presented on the zoonosis side, the COVID testing team, SRUC, and our colleagues at NHS that we've been working with. And thank you very much, and I'm very happy to take any questions.
Thank you very much indeed, for that fascinating insight into One Health, and really again demonstrates the breadth and depth of the work going on at the Moredun. A couple of questions have come through on the Q&A set, and I'll just field those to you now, Lee. Are there any plans to test the E. coli vaccine in the field in the UK as well as in the USA?
Sorry, can you hear me? I've just unmuted. I hope you can hear me all right. Yes, it's my colleague Tom McNeely who's leading that work, and the research is going on, as you say, at the moment in the USA. I'd have to check with Tom whether there would be plans to take it into the UK, but I would think if it was successful over there, I would think it'd be a very positive thing to bring into the UK. And it was quite an interesting one, because normally we would develop vaccines to help protect livestock, and I think it's interesting that this vaccine has been taken up in a way commercially, because it's a really good way, I think, of also a One Health approach to protect public health in particular.
Okay, and I think we'll just stop there, and Lee, there's another question in the chat. Maybe you could answer that on the chat forum, because time is moving on, and we will now move on to our final speaker for this morning, who is Dr. Hannah Shaw from Harper Adams University. So Hannah completed her PhD with the Moredun in 2019, so a very close link between the two institutions here personified in Hannah, and her PhD examined the production impacts, the disinfectant efficacy and the epidemiology of the parasite Cryptosporidium. And Hannah's research has led her to provide practical recommendations in the form of knowledge exchange events and newsletters to UK farmers in order to reduce the parasite burden on their farms with the aim of increasing and improving both animal welfare and farm profitability. And Hannah has been a lecturer in animal sciences for 3 years now, and she's only a few weeks away from her 1-year anniversary at Harper Adams, and I'm delighted to have her as a member of staff in my department. And Hannah's talk today is entitled From Moredun to Harper, a focus on livestock health, and we hand over now to you, Hannah.
Brilliant, thank you very much. I think I've just unmuted myself, so hopefully we can all hear me. Fantastic. So thank you very much for that introduction. I would like to take this opportunity at this time to thank the 3 speakers that we've had so far from Moredun. They've been really excellent. So both Stu, Mara and Lee, those have been absolutely fantastic to listen to so far. And also to thank those sort of in the background as well. Beth, Hazel and Blair, who've been sort of fielding the questions. So if you do have any questions as you go, please do feel free to pop those in the chat. Okay, so just one more sort of quick thing from me before I get started is if you have been unable, if you're an amateur member and you've been unable to sign in with your name and it still says anonymous, make sure you pop your name and your SQP number in the chat so that we can validate your attendance. Okay. So I am very aware that we have a multitude of people attending today's event. So not only do we have farmers, vets, SQPs, but we also have quite a number of students who are going to be sort of checking in today and having a look at some of these interesting talks. So I'm going to have a sort of range of things which I'm going to be talking about. The first one being an introduction and a sort of general career pathway, because I've chosen the sort of PhD route and then going into teaching. And I wanted to talk about that a little bit, just to sort of help give an idea of a career pathway for some of the students who happen to be watching today. I'm then going to move on and have a look at my PhD focus. So I'm going to be looking at part of the research that I did at Moredun on cryptosporidiosis, and then I'm going to focus on some of the current research which is going on at Harper Adams. So a little bit of an introduction to me, just as a little bit of interest for some of the students who may or may not know me already. I did a Masters in Science in Veterinary Biosciences at the University of Glasgow. And this included a 12-month industrial placement. And this placement was really great for me because I wasn't really sure what I wanted to do. Initially, I was thinking of doing veterinary medicine as a second degree, but I wasn't really sure what other options were out there regarding animal health. And so I did this 12-month placement at the Moredun Research Institute. And it was really excellent because it allowed me to develop that real world experience, to develop my confidence and also to allow me to know better what sort of potential job opportunities were actually available other than perhaps moving into veterinary medicine, as some of these other opportunities aren't necessarily as well advertised. So through networking, it was possible for me to sort of realise what it is that I really wanted to do and how much I really enjoyed actually working at Moredun. And while I was doing this placement, I was working on a couple of protozoan parasites, both Cryptosporidium and Toxoplasma, and also looking at their epidemiology, how they move throughout the environment. And we also did a sort of whole catchment study, also looking at public health risk as well. So after I'd done that placement, I was invited back to complete my PhD. And this was done at the Moredun Research Institute with the same team and also in collaboration with the University of Edinburgh. I then, following my PhD, decided to go in and become a lecturer. So I've been a lecturer for about 3 years now, starting off at the University Centre Rees Heath, and now I've almost been here almost a year at Harper Adams. As far as actually choosing the PhD route rather than deciding to go on to do a second degree, really it was that industrial placement, which is offered on all of our degree programmes here at Harper, which really sort of gave me the idea that actually I could do, there were some other opportunities and other things that I could do. So I was really keen, because I had that sort of level of knowledge from the industrial placement, to further that knowledge and understanding of infectious diseases, as that was what I found really interesting. Not only that, but I was very interested in doing very practical work. I enjoyed being hands-on. And actually, there are quite a few PhDs out there which allowed me to go out onto farms and sort of still have that level of that sort of practical element. And so that was very interesting for me. It allowed me to develop that sort of level of independence, and also to be creative with a project that I could make my own. Probably not as big a thing right now due to COVID, but there was quite a lot of travel involved as well. So I was able to go about, we went to places like Madrid and Cuba, where I was able to present my data and talk and network to a wide variety of scientists, which was very exciting and interesting. So I know we have quite a few of our sort of final year students who are watching today. Maybe they might consider going in to sort of further research, whether that be a master's in research, whether that be a PhD, and I would definitely recommend it if that is what you're interested in. But let's move on now to sort of highlight some of the research in which I did during my PhD on the parasite Cryptosporidium. So Cryptosporidium is a protozoan parasite which is transmitted via the fecal-oral route. There's just around 44 recognised species, although there very well could be more by now, and there are many more genotypes. Some of these are zoonotic, so it can go from animal to human, and so therefore not only is it a problem for our livestock, but it also creates a public health risk as well. It's associated with human outbreaks through recreational water contamination, so swimming pools and such like, and also through drinking water. The main symptoms of cryptosporidiosis are watery diarrhea, which is also referred to as scour, abdominal pain, loss of appetite, and in some cases where perhaps the animal has not been rehydrated quickly enough, it's not been caught on time, maybe they have a weakened immune system, maybe they're very young, they could have concurrent infections, it could actually result in death. So I specifically looked at Cryptosporidium in cattle, and typically there are different species which are found in cattle, but each of them have a sort of different clinical manifestation of the disease. So typically very young calves, so that's those that are from birth through to 6 weeks of age, they tend to be infected with the species Cryptosporidium parvum. And the clinical signs of disease in which we would see for this are diarrhea, loss of appetite, abdominal pain, and a mild fever. As these calves sort of grow up, then they are sort of infected with other species of the parasite. So we have Cryptosporidium bovis and ryani, and then as they get a little bit older, we also have Cryptosporidium andersoni as well. Although part of my research did find that Cryptosporidium parvum was actually present throughout all of these life stages. However, when they get a bit older, we don't tend to see those clinical signs of disease. So Cryptosporidium bovis, ryani, and andersoni, they are not really sort of associated with clinical signs. There are a couple of studies that show things like reduced weight gain, reduced milk yield when they are adults. However, in general, it is those sort of younger animals which show those severe clinical signs of disease. This is also the case in sheep and lambs, so it's our lambs that tend to suffer with severe clinical signs, showing sort of very similar, so diarrhea, loss of appetite, etc. However, our other species which are found in sheep, so Xiaowai, ubiquitum in the slightly older lambs, and we have Xiaowai parvum and ubiquitum in the adults. you don't tend to see, or there hasn't been any published research showing clinical signs of disease in these animals. With regard to transmission and how they actually become infected, it's thought that there are multiple transmission routes, or there are multiple theories with regard to transmission routes anyway. So this could potentially be from their mothers, which was part of my research to be able to analyse this link between dams and calves, from the environment, from other animals such as wildlife, and then also potentially from their drinking water. And the main problem is that these ursus, which is like the infective form, is that they are very environmentally stable, which means that they're resistant to many of the commonly used disinfectants, which we would use on our farms. They're resistant to heat, so up to about 60 degrees Celsius, and cold down to -20. So as you can imagine, very difficult to remove from the environment. And they can survive up to 12 months in cool, moist environments. Not only are they very environmentally stable, but they have a very low infectious dose. And so infection can spread very rapidly through a farm or groups of animals. So it's been found that as few as 17 ursus are required to cause disease in calves. However, once that calf becomes infected, it is able to shed billions of ursus into the environment, which are very environmentally stable, difficult to get rid of, and so easy to spread throughout that group. So how does this actually happen? So a very quick life cycle for you. These ursus are ingested by the calf via the fecal oral route. And in the gut, the ursus will hatch and it will release 4 sporozoites. Here they will develop into infective ursus, where they're either shed out in the feces, ready to then infect sort of surrounding animals, and also some ursus will remain in the gut to reinfect the calf. So they only require about 17 to actually become infected themselves, and then they will shed billions into the environment. So you can see it's quite a problem. Another reason why it's a problem is because there are limited treatment options available. So there's no vaccine to prevent disease. However, certain antigen targets have been detected, and so there are some promising opportunities maybe in the future. There's no licensed products for the treatment of lambs. And there's only one licensed product for the treatment of calves, which is Halicure. This is a good product, but it does reduce the shedding and it does not cure the disease. And so it may be that you're reducing the clinical signs of disease, but they're still shedding ursus, and with such a low infectious dose, they can still be a risk to the other animals around them. It is potentially toxic if it's not used properly, so if it's given at twice the recommended dose, and it cannot be used in scouring animals already. And it has to be used for 7 consecutive days from birth, which can be quite difficult depending on your farm setup. So the aims of my PhD were to examine the role of adult cattle in the transmission of C. parvum to their calves, to determine the role that wildlife plays in the transmission, to have a look at the available disinfectants which we have and determine which ones work and which ones are the best to use, and also to determine whether or not C. parvum affects the long-term growth of beef calves. So the one I'm going to be talking to you about is looking at the economic impact and looking at this long-term growth. So the big question here really was, does Cryptosporidium affect the growth rate of beef calves? So it has been found that there is sort of damage to that gut lining. However, the sort of rate of recovery has not been sort of properly documented yet, although it is still, there are research projects having a look at that sort of gut recovery rate. It has been found that both Cryptosporidium and Giardia are associated with reduced lamb carcass productivity. Not only that, but it's able to sort of result in growth faltering and reduced catch-up growth in children as well. So previous studies do show that there is a potential for it to actually have an impact if they were to be infected at this young age, that it would have an impact sort of later on and therefore cost farmers quite a bit of money. So in order to determine this, we chose a farm which had a history of clinical cryptosporidiosis. So previously, this farm would suffer anywhere between 15 to 30% of calf losses due to this parasite, but found success with improving colostrum quality, which then resulted in a range of clinical manifestations of the disease. So we had some which were shedding oocysts, however, were not showing clinical signs of the disease. And then we had others which were sort of really very sick. So we had that spectrum. So we were able to analyse then, put these animals into groups and sort of follow them throughout their sort of 6-month period until they went to market. So we chose this farm because it allowed us to compare those with clinical disease to those with severe clinical disease. So no clinical disease to severe clinical disease. So this farm was visited every sort of second day for about 4 weeks. And this was between sort of 0 to 16 days of age for the calves. And then these were then compared to the animal's weight, which was weighed at 4, 5 and 6 months of age. But in order to do this, we needed to create a scoring system. So not only did we score their fecal consistency, so one of the biggest clinical signs of disease is scour in these calves. So we would have a look at their fecal consistency and provide a score based on that. And then also have a look at their demeanour. So how they were acting, were they standing, happy to rise, were they sort of inquisitive, or were they actually unable to rise? Were they dehydrated? Did they have a low body condition score? Those sorts of things. So we created this system, this scoring card for cryptosporidiosis, so that this could then be used to provide scores to all of the calves on this farm. So here we've got some images of what a high scoring animal would look like. So you can see that sort of ear droop. You can see that they've got a sort of at their rear, it's quite wet where they've had the scour. We've got some sunken eyes in these images here. So you can see sort of what this looks like in high scoring animals. So they were split into 3 groups. So we had severely infected, mid-range, and then those with no clinical signs of disease. So if it was clinically severe, that covered a sort of watery diarrhea for an extended period of time. So that might be 3+ days. And they had to have a poor demeanour and also be reluctant to rise. All the way down to that no clinical disease, which covered animals which had a score of probably 0, to those which may have had maybe a score of 1 in their demeanour category on one occasion. So they might have had a slight ear droop or looked a little lethargic on one occasion, but they didn't have any scour. So here you can see the sort of number of calves that we had in each group. After this, we had to make sure that they weren't infected with anything else. So this was both during the study and sort of in the period while they were being weighed. So we had sort of further gastrointestinal pathogen testing. And this included E. coli, rotavirus, coronavirus, and the farm is also VVD free and Johne's free. So we weighed these animals both at birth, first at birth, and then we had 4 months, 5 months and 6 months in order to have a look at the differences which may occur between these different groups. So here we have a graph of the results. So the sort of top line that you see here, these are our low scoring groups. So these didn't have any clinical signs of disease. And then we have our high and medium. They're crossed together quite closely, so you can't really tell the difference between the two. But that's actually really interesting, that even those with a mid-level of range of infection still had this decrease in weight. And this seemed to occur very early, so it occurred even at that first weighing point at 4 months, where you could see the difference in weight, both at 4 months, 5 months and 6 months. So even those that had just a mid-range level of infection still showed the significant difference between those and the low-scoring groups. As far as putting this into cost and how much this could cost for a farm, the direct losses associated with this reduced weight gain found on this farm related to sales of approximately £130 per affected calf. However, we do have further costs as well. So we have increased feed, husbandry costs, additional labour looking after sick calves, potential treatment, veterinary costs. So it could actually be quite a bit more than this £130. So that's just for the reduced weight gain related to sales. So it is a significant economic burden to the cattle industry, and this work was published in 2020. With regard to how to actually manage it, because obviously it is quite difficult with these oocysts being so environmentally stable, is we need to be able to reduce that environmental contamination. So steam cleaning of animal pens, calving areas, cleaning calving areas frequently, deep and regular straw bedding, and then also our animal management. So the 3 Qs of colostrum, quickness, quality, quantity, keeping animals in age groups, so trying not to mix older animals with younger animals, quarantine any sort of scouring animals, and also to be able to feed and treat any ill animals after the healthy animals. So I now would like to sort of move on to the final part in the last few minutes that I have to discuss the current research which we have going on at Harper Adams. So we have plenty of industry funded work on livestock health covering sheep and lambs, cattle, pigs, poultry and aquaculture. We have an Innovate UK funded project which is looking at precision farming in the dairy sector, and this is in collaboration with China. We also have an AHDB funded project which is looking at dairy cow housing. And we're also partners in a Horizon 2020 EU funded project looking at the welfare of poultry and pigs, which are reared in organic and low input and outdoor production systems. I would like to sort of have a little bit of a closer look at a research link which we currently have with the Moredun Research Institute. And this is on aquaculture and aquatic animal health. So the aquaculture research at both Harper Adams and Moredun Research Institute focuses on sustainable disease control strategies, and this link was established in 2016. Since we've had this link, we've had student research projects, we've had further development into aquatic vaccines, and we've had some scholarly writing, so sort of 3 book chapters on the viral diseases of Atlantic salmon, and also the publication of peer-reviewed articles. And the scientists which sort of play a key role in this are both Kim, so Dr. Kim Thompson from the Moredun Research Institute, and we also have Tharangani from Harper Adams. And their current research focuses, so we've got Kim's focuses is development of vaccines for cold and warm water aquaculture fish species, including vaccines for sort of sea lice. We've got cardiomyopathy syndrome related research, complex gill disease research, immunology studies and pathogen identification. And then Tharangani here at Harper, current research focuses are alternative therapies for controlling AMR. We have vaccines for early life stages, gill health, and then novel immunomodulatory feed ingredients as well. And having links is really important because we have facilities in both places which are useful to the other groups. So here we have a list of the different facilities which are available both at Moredun and also at Harper with regard to aquaculture. I won't read through all of these as I'm short on time, but it might be interesting just to have a quick look and just to sort of appreciate that actually these collaborations are very, very important in order to share the facilities and further the research in these areas. Focus for this group is being able to seek collaborative research funding. This includes sort of joint research studentship projects, so that might be future PhDs or Masters in Research, or potential for student placements. Not only this, but they're hoping for this collaboration with the Moredun Scientific for industry-led commercial research and RAS system. So feed additives and novel feed materials, veterinary vaccines and medicines, biocides, seed stock and equipment and devices. I would like to focus on one of our PhD students as well. So PhD students, we have quite a few PhD students at Harper, and they're some of them working in the area of livestock health and welfare. And this is funded by industry, AHDB, and also various charities as well. And one of these students that I'd like to focus on has had a recent publication and also another one in review, which is Caroline Best. And she's looking at, and her research focus is lameness in sheep. And this is, the first published study was a cross-sectional observational study of 532 UK sheep farmers, documenting the uptake of a national lameness control plan. And then there's a second paper, which is in review, which is influencing the sort of the factors influencing the farmer uptake of the 5-point plan through these qualitative interviews. And this is highlighting a need for increased veterinary farmer collaboration when implementing long-term lameness control plans. Any sort of research in which we do like to share this quite a bit, so via our social media sites, so via Twitter, via Facebook. We do quite a lot of knowledge exchange, especially where we have some new and exciting research, or we have some new publications which we'd like to share with you. So one of the recent ones was a sort of very popular article by Professor Michael Lee, who's our Deputy Vice Chancellor, and he's also a professor in sustainable livestock systems, looking at why a balanced omnivorous diet could help us save planet Earth. So this was shared via our social media platforms and was very popular. So you can see here we've got sort of 95 shares on that already. And this is a very, very interesting article, but anything like this, we do quite like to share our staff, our student expertise via our social media sites. So if you are interested, please do follow us on these platforms. I've got quite a few people that I would like to thank, so especially for webinar organisation, we've got Beth, Hazel and Blair, who are sort of behind the scenes, Philip for chairing, and also Amtra for sort of agreeing to provide the CPD points for this particular session today. And there's also plenty of other people that I would like to thank. I won't list them all now, but this has been a big collaborative effort, and I also want to thank you all for joining us today.
Thank you very much, Anna. Okay, so that was a fascinating insight into your pathway into research, and as you pointed out, and as has been pointed out by Taryn in the Q&A session, it's excellent for students to consider a research career pathway, and you're a classic example of that. And there's a question that's come through here from Connie. Did you have opportunity to choose the subject for your PhD, or was this an area that's Moredun already wanted to research? So we can make that a wider question, say, how does this process work of choosing a PhD and finalising the topic?
So that's a really good question. For me personally, my PhD was a sort of progression from a previous PhD. So they had, before me, a student, Dr. Sarah Thompson, who completed her PhD, and the sort of questions that arose from that one then resulted in the sort of questions which I was then going to answer for mine. I was definitely keen to go back to Moredun, so I was sort of following the sorts of PhDs that they had available. However, that particular one which I had was funded, so it was funded by AHDB Beef and Lamb, and also AHDB Dairy. And there was also some funding from the Scottish Government as well. So that one was funded already. We could sort of apply for that funding, and then the sort of student was interviewed. However, there are other opportunities where if you already have an idea, or you already have something that you would like to research, finding those who would be willing to sort of supervise, and then actually going for a different funding route. So perhaps scholarships are available, or you can go actually for funding yourself. So there are multiple opportunities, not only just searching for ones which are already available, but there are ones in which you can sort of have a little bit more input on the topics as well.
Okay, thanks, Hannah. You mentioned about the importance of your industry placement year. And what tips would you give to maybe students who are maybe in the early years of their degree course and about to go on placement, and also to employers as to the benefit of taking students for a placement year?
So the first part, as far as tips for students actually going out, if you are offered opportunities to do things, definitely take them up. They're not expecting you to be perfect in the first instance, and it is a learning curve, and you're there to develop that real world experience. You're there to develop that confidence and to develop those skills, and the employers are giving you that opportunity to actually do that. So say yes as much as you can, would be my biggest hint, would be my biggest sort of piece of advice. Even if you feel nervous, give it a go. And with regard to employers taking on students, not only is it sort of one very big interview process where you're essentially sort of seeing this student for 12 months, you're seeing how they're progressing, how they're dealing with new situations, new scenarios. You may end up with a fantastic new employee at the end of it, where perhaps you may want to offer them a job at the end, and it may well be that they're providing that extra pair of hands, that they are useful to you sort of within within your sort of industry. So yeah, I would definitely recommend both for students to choose the industrial placement and for employers to take on students as well.
Okay, thanks so much, Hannah. That was excellent. Thanks for that fascinating talk to round off our 4 presentations. So we now move into our panel discussion section of our event today, and I would invite Stuart and Lee to come back and join Hannah and Mara, who I can see on the screen. And for you in the audience, if you want to put your questions into the Q&A forum, please put the name of the speaker that you want to address it to first, to make it easier for me to pick up. I can see questions coming through already, and it's going to be a challenge for me to keep on top of all of these, so apologies if I missed your question, but we'll try and do the best we can. And Stuart's been answering busily as well, so thanks for the speakers for their answers on the forum. Can I kick us off, Stuart, with a question to you? We've known about sheep scab for so long, so many years. Why do you think we're still struggling so much, it seems, to try and control this disease?
Yeah, it's a good question. Yeah, I mean, I think partly, we got very close to getting eradication of scab, but ultimately we didn't maintain, and we got it down to very, very low levels, and we're now back probably in the worst of days in terms of where sheep scab is. I guess it's partly that first of all we haven't had a test in the past, and that certainly helps. But equally we don't have as many treatment options as we have had in the past. So we are in a difficult place. We need to coordinate, I think, is the biggest issue, and that's partly what this new RDPE funded project will aim to do, and also partly what we tried to do on Mull as well, which was to take that sort of regional approach, demonstrate that you could use the test to find scab when it was there and then treat it accordingly, and then also to come in with best practice advice so that you leave a legacy, so that you're not just going in, sorting out what's there, firefighting, and then walking away. And I think that's kind of really important. So this new project, what we're doing is identifying hotspots. We're not shying away from this. We want to know where scab is. We want to go in and target some of the worst areas for scab. But we still have to build a defined cluster within these regions, because we have to have some boundaries, and that can be a geographical cluster. It could be bordered by roads or by kind of hills and other sort of factors. Mull obviously was easy because it's surrounded by water, but they're not always that easy, and we've had previous eradication campaigns in the Pentland Hills just outside of Edinburgh, and also in the Angus Glens further north. So we've shown that we can do this. I think that this is a really big development because we're taking 3 significant areas, all of which have big problems with sheep scab, and we're going in, working as closely as possible with the stakeholders in those areas. It's a boots on the ground exercise, so we have people there to coordinate, that will work with the farmers, coordinate the vets as well, and then try to actually get control. And I think if we do that, show that we can be successful in doing that, that will then seed us moving into other areas. And eventually, you know, through that sort of approach, I think we can bring scab down. Whether that leads to an eradication, we'll just have to see. It depends how successful we can be on that regional approach first.
Okay, thanks, Stuart, for that excellent answer. A question has come in from Annette, and I'm guessing this is directed at you, Hannah. Is there a good recommended industrial steam cleaner for farm buildings? And maybe we could widen that a bit and say, you know, how important is cleansing and disinfection in terms of biosecurity and animal health at farm level?
Okay, so very good question. With regard to an actual steam cleaner itself, I'm afraid I'm not familiar with different brands. However, Crypto can survive sort of up to about 60 degrees. So you do want to make sure that it is actually covering that, especially when it's actually hitting the bottom of the pen, that it is at that temperature. Not only that, but you need to make sure that those pens are cleaned out properly, first of all, as that ensures that there isn't any leftover oocysts which are hiding in those nooks, crannies, anything like that, or any sort of fecal material that's being left over. So in order to ensure that that steam cleaner works effectively, you want to ensure that, first of all, it's properly mucked out and cleared out as best as possible, and that you're using that steam cleaner that will get up to the required temperature in order to kill those oocysts.
Okay, thanks, Hannah. Mara, can we ask you, there's obviously lots of development of point of care testing for diagnostics, so that tests can actually be done on a farm or with the animals in situ. How do we get this balance between diagnostic laboratories and point of care testing? How would you like to comment on that?
So point of care diagnostics are very useful. They are generally cheaper than laboratory testing. They can be carried out by personnel that does not have to be as specialized like the one in the laboratory, in the labs, and they can give a quick answer. My only comment is that those will be expanding, but they will always have to be run in parallel, not for every sample, but maybe to confirm the detection of an outbreak or disease in the farm by laboratory testing. That will reduce the amount of sample that we go to the lab, so eventually we'll reduce the cost, but there are problems of sensitivity at some time of specificity with some of those point of care diagnostic. So a confirmation is always required, but yes, to try to reduce the cost of the testing to the farmer and to get a very quick response in case of an outbreak, those will be a very beneficial development.
Okay, thanks, Mara. There's a question that's come in relation to pen-side testing for scab. I'll direct this to you, Stuart. When might the pen-side scab test be commercially available?
Yep, it's a very good question. Again, I think we're very close. We have 2 different platforms that we've been working on. We're very close to having that available. It hasn't been as straightforward as we would like, and partly, you know, this is down to the issues that Mara just commented on the reduction in sensitivity that you have with these types of tests, and partly that's down to the fact that when you run a sample on a point of care test, what you're interested in is speed. So of course the tests run more quickly. And in our case, what that means is that there's less time for the antibodies in the blood to react with the antigen on the test and give us a test outcome. And that's what we've been dealing with the sheep scab point of care test, is that reduced sensitivity and balancing the performance of a test in the field, where you don't really want the test to take longer than, say, 20 or 30 minutes. Ideally, the test should take only 5 minutes. So it's trying to find that balance between sensitivity, the time that it takes the test to run, and obviously, you know, the sort of sensitivity and the sensitivity balance for that. So we're not quite there yet, but I'm hoping that we'll see some developments in that field during this year. We would have been further ahead last year. Unfortunately, COVID has interrupted pretty much everything. So yeah, so hopefully this year we'll see some more developments in that area.
Okay, thanks very much, Stuart. Can I direct a question to you, Lee, please, and bring you in? You talked about interdisciplinary collaborations and research, and I've been involved in interdisciplinary research myself, and it's something that fascinates me. There are obviously lots of positives. Have you found any challenges in working in an interdisciplinary format?
That's a really good question, and I can tell by the question you've done this before. But yes, I think it's one of those things that when you first move into it, it's a little bit scary, because you're actually interacting with people who have quite different perspectives from you. And then once you get the hang of it, it's really quite fun. And if I can give an example of a workshop that we were all involved in a few years ago, and Stuart was involved in this one, where we brought together biologists, scientists at Moredun, and engineers from Heriot Watt. Mara's probably involved in this one too. But really, what we did this sort of speed dating, where we basically all sort of shouted at each other and told everyone what our interests or issues were. And at that time, we hadn't developed any of these pen-side diagnostics, but we said to the engineers, what we'd really like is to have a rapid diagnostic. And engineers, being engineers, went, oh, well, we'll sort that for you, because they're engineers and they're used to sorting out problems. So they basically went off and I think worked together with Stu. They got a grant, and that really helped to push forward that pen-side diagnostic, because you were dealing with people who thought very differently from you. Now you're talking about challenges. Challenges are they think very differently from you. So sometimes the same thing can work against it, because people have got very strong views on how things should go forward. So I think, sometimes the challenges are also if people have disagreements and they think there is a better way forward. I think a good example of that is if you go on the BBC News at the moment, and you can see all the differing opinions from all the experts on how to deal with COVID. I think it's maybe quite a good example of how do you sort all that out. But I would say to people who are, are interested in having a go at this, you will gain so much from reaching out to other people in very different sectors. So I would say to all the students who are on the call, don't be afraid of doing this. You will learn so much from other people. And the way we do science often is we are trained in our disciplines, and with that comes you can get stuck in your discipline a bit. And I think if you have the courage to come out of that, you can often get really good solutions. So sort of a long-winded way of saying, Philip, I think there's more advantages than disadvantages.
Yeah, thanks, Lee. Absolutely agree with you. I took that to heart and went off and did a PhD in geography. I'm a vet, so that was definitely stepping into a new discipline, but I found it absolutely fascinating. The challenge for me is geographers speak a different language to vets. I had to learn the language of geography, but it's been fascinating. I've had a fascination with social science research even before that, but I absolutely agree that there's so much to be gained from interdisciplinary work. Excuse me. So a question for Hannah. Is there any evidence of resistance to Cryptosporidium in specific cattle breeds, or is it accepted that all breeds can be infected equally?
Oh, very good question. As far as cattle, I'm not aware of any research which is looking at different breeds, although I'm not sure if Lee knows of anything. I know that there has been a couple of studies in sheep where they've had a look at different sort of sheep breeds, and they've not found any difference between the breeds. So I would imagine probably not, but we don't have the research to be able to answer that question as far as I'm aware.
Okay, thank you, Hannah. Stuart, I'll direct this question at you. Carol Ann says there's a problem on the hills between Herefordshire and Wales with common grazing and a lack of coordination between farmers. What are your suggestions? How do we get farmers to work together to control scab?
Yeah, so in some way you've sort of answered the question, which is coordination. And again, I would refer to the RDPE project, but unfortunately that will not work across the border. So again, it's not just coordination at the local level, it is coordination nationally, and between devolved nations, because if we don't have that coordination, sheep scab is like COVID, it's not interested in borders. So, we place these artificial borders there, we make it more difficult for ourselves. So the key is coordination. The key is, yes, clearing commons for a prescribed period of time, working with the local authorities in order to achieve that, and where possible using the diagnostic test. If that is going to help in these scenarios, then yes, please do use it. Remember also that the diagnostic test can be quite powerful in these situations because it can pick up scab. So let's say there's scab on the common, you don't have to test everybody's animals. You can just test a handful of those animals, and that will act as a sentinel for everything else that's going on the common. So the test can be used in that way also. But ultimately, it comes down to how do you get rid of the problem, and that is coordinated treatments, clearing the common and coordinating treatments, and then trying to work out how it got in there in the first place. So a little bit of sort of detective work to prevent that happening again in the future.
Okay, thanks so much, Stuart. Mara, can we go back to you, please? And you mentioned doing diagnostic work in grouse. And could you comment on how important it might be to look at wildlife diseases in the context of livestock health?
So often diseasing wildlife is not looked at as much as it should probably be, because most of those species could actually act as a sentinel for the presence or for a spread of the disease. For example, we are looking, I didn't have time to show you this morning, because we are interested in tick transmitted diseases, with the fact that the ticks are expanding their habitat, their range, and their resistance to different climatic temperature and conditions. We are looking in deer and as a sentinel for presence of louping gill, Lyme disease, and Babesia and Anaplasma. And deer often have contact, especially in Scotland, with livestock species, the ones that are raised not intensively. So if we can detect area in which we can use those samples, either the ticks or the deer that are culled, and we get samples from those to monitor disease in specific area, that will be beneficial for, will give information to sheep farmers, but also will be beneficial for human health, especially in regards to Lyme disease. But this is just an example, yes.
Okay, thanks, Mara. Please do send your questions in, and remember to put the speaker's name at the beginning so that I can direct it in the right person. Lee, can we go back to you, please? And obviously, the whole theme of your research is One Health. What do you see as the future of One Health research and development in this area?
Oh, that's a really big question, yeah. I mean, I actually think that because of the COVID situation that we're all in at the moment, we're all in a big One Health experiment just now. And I think a lot of the, if you think about this time last year, how many people in the population knew much about, R numbers and vaccines and epidemiology and transmission. And if you think about it, we all know about this now. We're all discussing and debating it. And the other things that are coming up are, how did this occur in the first place? What's happening in our planet? How are we all using natural resources on our planet? And it's opening wider debates about climate change, loss of biodiversity, how that's affecting not just our health, animals' health, but the planet health. So I actually think this is, going to really open up lots of interest, debates, discussion about One Health, because we're all living it just now. And I think for, think about children growing up in this, and actually looking to scientists are now the new superheroes for children. And we'd never have thought that a year ago. So I think it's a really, in some ways, a very positive time for people to have discussions about One Health, because it's really brought home to a lot of people is we are all responsible for what happens on our planet. And if you look to the future with our children, you want to leave the planet in a better place. So I think a lot of those really big discussions are now taking place, probably in the back of COVID, because it's focused us to really think about that. So that's a big answer. And a small answer is that we are very keen at Moredun to adopt this One Health approach. And I think going back to that project we were doing with the NHS, which came out of that, us all trying to step forward and do something in the pandemic, that's going to open doors now for us to collaborate much more closely with NHS. And it's something that we've all been doing little bits of, not just Moredun, but across UK. But I think this has been rapidly accelerated because of the crisis, and that has been a really positive thing. Because as Mara and others have pointed out, these diseases know no boundaries. It's not like they say, oh, I know now I'm in Edinburgh or I'm in Wales, or no, they don't. So we need to work across countries, across the world. We need to share our knowledge. We need to all work together. So that whole thing about collaboration has really come to the fore with COVID.
Okay, thanks, Lee. And as a vet, I've always been fascinated in this overlap between animal disease and human disease, and it's something that we're trained to think about. And certainly when I was working in practice and in government, zoonotic diseases that have been the focus of your research, and particularly have been very much at the forefront of my mind. And it's a vitally important part of research and development. And I think that, as you say, that we've now opened up to the public and shown the power of science, and to think about the interaction between the food that we eat and the animals that we keep, and not just our pets, but also our livestock. And there's so many things to investigate and to understand better. And Hannah, can you just comment, just following on from that, you mentioned about knowledge exchange events coming out of your PhD research. How important is it for science to be put out there and taken out of the laboratory and brought into the public arena, in your experience?
Oh, absolutely essential. It's absolutely essential. I mean, the gap between us actually sort of producing results and getting that into a peer-reviewed journal can sometimes be a year, or it could be even longer. So actually being able to get that information out sort of as soon as possible, so that there may well be sort of actionable consequences from the research which has been done. We can actually sort of start sharing that sort of even earlier. So even with sort of PhD research, not only are you sort of getting that opportunity to sort of practice presenting it, but actually a lot of research which is being done by master's students, PhD students, is really relevant to multiple different sort of areas. It might not necessarily be that sort of one section in which you are part of, and actually that can then sort of simulate more ideas where somebody will sort of take your idea and continue that in a different area. And that wouldn't be possible if we didn't have these conferences, if we didn't have sort of times where like-minded people all got together and sort of shared these ideas and shared these results. And not only this, but being able to actually share this information with farmers, with vets, with SQPs, with students, with the general public, is also really important, because it is those people that are actually going to put this research into practice. So we're the ones maybe doing the research and providing the results, but you're the ones who are actually going to take that information and provide that advice. And if you're a farmer, actually sort of use that information on your farm. So it's absolutely essential we get that out there, because without that, then our research doesn't mean anything. We need to have sort of actionable consequences from the research in which we've produced.
Thanks, Hannah. I mean, a theme that's come through again today, and it came through in Stuart's talk in particular, is about resistance to animal medicines. I'll just open this up to any of you. You know, we have this challenge of antimicrobial resistance, and I use that in its widest sense of the term to include antibiotics as well as antiparasiticides. I'm very conscious that we have SQPs and vets in the audience today. What comments would you have on how your scientific research has contributed to these grand challenges of trying to prevent or to reduce the speed of development of antimicrobial resistance? And I'll open that to anyone who wishes to comment.
I can kick off and then pass to the others if you like, Philip. But this is a huge problem, I think, for everyone working in this area. And we know that a lot of the medicines, antibiotics or antiparasitics, there's not a lot of new ones coming out, so we have to really conserve the ones we have. And in doing that, you need your diagnostics to work correctly so that you know what disease you're tackling and you're focusing the medicines where they're needed. But equally, we talked a lot today about vaccines and that whole thing about prevention being better than cure. And I think a lot of the work we're certainly involved in Moredun is developing vaccines. So we're looking at how do we actually prevent these diseases in the first place. And again, it's also back to a One Health thing. It is what happens to the medicines you're using. They should be used responsibly in terms of used to actually prevent disease, not as a sort of prophylaxis. You can see this in malaria, where people have used malaria as a prophylaxis and then there's lots of resistance. So we know that all the bugs in the world will eventually become resistant to the drugs we've got. So we need to be cleverer about how we apply vaccines or apply our knowledge of epidemiology and diagnostics to target that. But I know there are others, probably particularly Stu has got experience of this. I don't know, Stu, do you want to comment on, take that a bit further?
Sure. Yeah, I mean, I think, you know, with Moredun Foundation last year, we produced a booklet. I mean, this is specifically looking at the sort of anti-parasitic compounds called Test, Don't Guess, you know, which is all about, you know, kind of testing first and making sure then that by doing that test, getting the specific diagnosis, that you're giving the right treatment to the right animal at the right time. And I think that's really important. So, you know, so as a prescriber, you know, you should be asking those questions, you know, with your clients, you know, is this the correct time to give that particular treatment? Is that the best treatment that I can give at this time? And also, what are the other issues? Is there resistance in that flock, for example? Are we having to manage around that rather than just, you know, kind of giving the same treatments again and again, you know, regardless of efficacy? And then I think, you know, the sort of points where Lee's made about vaccines, we all have to be conscious that there isn't a big pipeline of new compounds coming through that we can keep throwing at these, and they are very, very clever. You know, these organisms, particularly the parasites, which is I put my hands up, they are my interest, my passion. You know, they're very good at developing resistance. They have high population numbers, rapid growth rates, and they will eventually select for those mites that have developed resistance to particular compounds. And it's those then that we're left to deal with. The good thing about a vaccine is, I mean, I'm talking here particularly about some of the vaccines that we're developing against these parasites, is that they don't tend to target just one aspect of the parasite. They're hitting multiple targets. And by doing that, it's much more difficult for them to develop resistance in the short term. It's not to say they won't develop resistance, because ultimately they probably will. But the key is not to drive that resistance as well. And again, that comes to prescribing the right compounds at the right time, because if you get that right, you're not constantly pushing them towards resistance. So if we don't do that, then we're not helping them to achieve their ultimate goal.
Thanks, Stuart. There's a good question has come in from Georgie here, and it comes back to communication and collaboration again. But and I'll ask this to any of you. Do you have any suggestions on ways of engaging how everyone involved in the industry can give a focus on biosecurity quarantine for issues such as anthelmintic resistance, fluke and scab. So quarantine and biosecurity, how do we get that message across into the industry to encourage action?
I'll butt in very quickly, Philip, and then I'll hand over to Lee, who really is the expert in this area. But I would say definitely take a look at some of those resources that Moredun and our partners have been developing. That Tests Don't Guess biosecurity booklet is a really good production, and I think that's definitely very helpful. But equally, we also have to think about how we communicate those messages. We all know what the messages are, but it's how do we get that across to you know, to the people that really need to be taking those up. And we have looked at alternative methods, for example, cartoon series, animations, just trying to get that engagement. And I'll pass over to Lee, because she really is the expert in engagement. So
just to say that I think it's something that Hannah said actually in her talk, that sometimes listening to the people you're trying to engage with really helps you. So finding out what they're interested in. And usually with farmers, it's there's an economic interest. So if you're giving them messages, you also relate to what they're interested in. So if you say, well, look, if you apply effective biosecurity or quarantine, you're going to save yourself so much money a year, they'll start listening to you. Or if you do it in conjunction with showing them, like maybe on a monitor farm, how you adopt this process and how much more efficient it is for them, they'll listen to you. In terms of what Stuart was saying, though, absolutely, we've got, I think Hazel is very kindly putting up some of the resources on the chat for people, but Stu was involved in some fantastic cartoons to help tell people about the sheep scab stuff that's coming through. We've also done some animations working with Selena Wagner, who's a super animator. Again, that was a good example of collaborating with people who are really good in the visual arts that can help with messages. But one thing I would say, I'll just go back to who are the people you're trying to talk to and what are they interested in, and then trying to work with them in that way and give them some good examples of how it's in their interests to adopt some of these approaches.
Lee, don't forget to mention the bus.
Oh, thanks, Mara. Yeah, thanks, Mara. This was a project we had for our centenary, and I don't know if any of us actually had the old bus in our slides, but normally at Moredun, if we give a talk, we've got the old mobile laboratory, which is when Moredun started off. There were about 2 scientists, I think, and they went around in an old Shepherd's hut and wheels, literally out round to do field work. So what we thought for our centenary was we'd update this, and we've got a brand new mobile laboratory, which was all set to do a tour of the UK last year. But as we won't mention COVID again, but COVID has put paid to a lot of our plans. But we have got this wonderful mobile lab and education outreach bus, and I really hope next year, Philip, we can bring it down to Harper Adams and maybe have it, Hannah, as part of one of the displays, and we can show people around. But the idea is really to take diagnostics again back out to the field, but also help us engage with farmers on some of the issues they've got and show them what sort of tests we're doing and really get them involved. Thanks, Mara, for that that plug. Okay,
we look forward to seeing the bus next year. A question for you, Lee, and I know that the E. coli work was more your colleagues at the Moredun, but the question is, has the E. coli vaccine potential for use in the control or prevention of diseases such as watery mouth in the neonate lamb, or should farmers place greater focus on strict pen hygiene and reducing environmental burdens?
Right, that is a good question. I think the E. coli vaccine is mainly geared at, obviously, for cattle and to prevent that, prevent shedding in cattle. So I'm not sure the potential there. I know that certainly very important to do your biosecurity. And I appreciate the point of the question there about lowering environmental burdens. I probably have to pass. I don't know whether Mara, I don't want to put you in the spot, but if you had, she's shaking her head. Sorry, I did. Our colleague who works in E. coli, I did text him a couple of times to see, but he's obviously busy, but we'll try and come back to you on that. I wouldn't want to give you the wrong answer there.
The thing I would add to that is the importance of colostrum, and yes. We
good
have
point, Philip.
had a PhD student who's looked at calf rearing, dairy calf rearing, and the importance of colostrum, and has published on that. And I have another PhD student at the moment, Paul Crawford, looking at antimicrobial stewardship in sheep in Northern Ireland. So we look forward to Paul's research coming out in the coming years. And we'll just have a final question there open to any of you, and then I will round up and close our event. But to all the presenters, is there any advice you would give to students, especially those starting their degrees?
If I may intervene, I would suggest take every single opportunity you have been offered to. In my example, I just finished a PhD. I was going to start a postdoc. I was offered out of the blue the possibility to go 6 months to Kenya to do some training in immunology, and I took it. I just went, yes, no problem, I'll go. And that changed completely, not only my career pathway, but also my life. So you never know what is out there. Don't be shy. And as Anna said, people will not expect from you to be perfect at the beginning. I do expect to be perfect towards the end, but that's just me. But just go for it. People will not judge you. People will appreciate the effort that you are making.
I think I'll sort of quickly add to that and essentially just say that you really do get out what you put in to your degree. So I think taking those extra opportunities, anything that is offered to you, any sort of extras, you'll find that within your modules, you'll get given sort of extra reading or sort of extra things in which you can have a look at. If you find something that's really interesting to you, follow that, do a little bit more research on it, because you never know where that could lead. You may think at this point in time that you know exactly what you want to do. However, I would probably guarantee that you're not necessarily aware of all the possible routes in which you could take, and there's probably a lot there that you haven't thought about. So do take those opportunities that you can, put in as much as you can, and it really could open up some extra options for you, would be my advice.
Thanks so much, Anna. So our time is almost up. All it remains for me to do is to sum up and just to refresh your memories on the journey we've been through this morning. So we started off this morning at 10:10 with Stuart Burgess looking at sheep scab, being reminded of the importance of this condition as an animal health and welfare issue, something we need to tackle. Stuart introduced us to the exciting research and development of testing and of vaccines. Then we moved on to Mara Rocchi, who talked about surveillance, the importance of disease surveillance, and fantastic work Moredun do in terms of diagnostic testing and also developing tests. And she looked at the respiratory disease, tick-borne fever, louping ill, and rabbit hemorrhagic disease, and also looking at the grouse as examples of the work that's going on there. Then Lee Ennis looked at One Health and talked about zoonotic diseases, focused on E. coli, but particularly on Toxoplasma gondii, emphasising the importance of this parasite as a One Health issue across the world, and reminding us of the importance of interdisciplinary collaboration and of science communication. And then we finished up with Hannah Shaw, introducing us to her own career pathway and route into academia and research, talking about the importance of placement in industry, showcasing some of her research on Cryptosporidium from her PhD, and introducing us all to the research that's ongoing at Harper Adams and the collaborations that already exist between Harper Adams and the Moredun in terms of research. And we've been through a fantastic journey. I tremendously thank all four of our speakers who presented excellent talks to us this morning. I echo the thanks of Hannah to those working in the background, and especially to Beth Wells and to Hannah for pulling this event together. Thanks to Blair and Hazel for recording the events and managing all the technology. I think everything has worked very smoothly and has worked according to time. Thank you to all of you in the audience for joining with us this morning, and I hope that you found this really useful and interesting and even inspiring, especially those of you who are students. And I hope that this will perhaps make you think about a career in research and the fantastic opportunities there. And there's so many questions to look at. The world is your oyster. So thank you once again to everyone, and do remember that it's recorded, so you can listen again and share it with your friends and colleagues. And I wish you all the very best, and thank you so much to everyone.
Thank you.
Thank